Hello, Madhurek. Welcome to the workshop. I'll give about, we'll wait until 10 past 10 and then we can begin. Okay, so the time now in the UK is 3 past 10. So we'll start around 10 past 10 UK time. Hello, Victor. How are you?
I'm okay, Prof. How are you?
I'm fine. I'm fine.
How is Manchester?
Manchester is always cloudy as usual.
And cold, I suppose.
Yeah, yeah.
Eh,
I miss Lusaka. I miss.
The weather is always nice.
Yeah,
Now we have some rains, but the weather is generally just nice. It's not
also.
cold, it's not hot, just nice.
Yeah, good.
The
Your family is fine.
family is okay.
Okay,
Yes,
good.
thank you.
Hello, Adam. How are you?
Hello,
Adam.
bro. I'm fine, bro.
Yeah, are you in Spain now, or where are you now?
Yeah, bro. From there.
Okay,
Yeah,
good.
yeah. Just on break. I said, let me join, and as you said that the program will start by 10 o'clock.
Yeah, we are waiting for more people to join before we
Okay,
start. Yeah.
okay. That's fine. No problem. That's
Mm.
fine. How is ATB? You are in ATB or at home?
I'm at home now today.
Okay. Today, no problem. That's
Oh
good.
my God.
Yeah, Kinsley, how are you?
Fine. How you doing?
Yeah, I'm good. Nice meeting you.
I too.
It's Nigeria.
Just do. Mm.
That's good. That's good. Now it's 11 there, right?
Yes. It's 11. Yes, past just a few minutes past 11.
Yeah. I'm sure you are 1 hour ahead.
Yeah. That's what. Mm.
That's great. So we are waiting for few people to join.
Yeah. Mm.
Okay. I'm also attending a conference here, but so we are just on break. I said, okay, let me join here first.
Okay. Mm. They're
Um,
in the UK or outside the UK.
yeah. No, outside UK.
Okay. Okay.
Yes,
Okay. Mm.
that's good. So the present, I didn't see the schedule. We have 1 hour or 30 minutes per presenter.
30
It's
minutes. Yeah, 30 minutes.
Okay,
20, and
30.
then 10 minutes for questions.
okay. That's good. So we wait for them.
Uh,
Thank you.
All right. Thank
okay.
you.
Well, welcome everybody. My name is Sarlis Nadaraja, and this is the third workshop on Black Heroes of Statistical Research. The first workshop was held in 2023. The second was in 2024, and this is the third one. It's good to see a number of you attending this workshop online. Um, the purpose of this workshop is to celebrate research about Africa, not necessarily done by African researchers, but this workshop is also open for anyone, not necessarily Africans, who are passionate about wanting to contribute to the African science and trying to help the communities there. So for the first time this year, we have a non-African who's presenting at this workshop today. Is Louis Bolton, who he'll be talking from 2:30 to 3 p.m. today. He is one of my students who's worked on a project with me. So this workshop is open for people of all colors, not just black, but white, brown, pink, yellow, whatever color you are welcome to attend this workshop and present your research relating to Africa and how it contributes, or how it will contribute to the continent, right, and the people there. Not just do researches for the sake of it, but do research because you want to contribute something to the people in the continent. So this workshop wouldn't be materialized without the help of Kinsley. She's been the backbone for everything I've done since for a long time now. Without her help, I wouldn't be able to stand here and talk to you. And so I must really, really thank Kinsley for everything, for all the help she's given me over the years and continued to give me. So she did most of the work for this workshop, as was the case with the previous workshops. So we are trying to, I think I might have mentioned this in earlier questions, but we are trying to work with as many people as possible in the continent, and trying to help them to achieve, what they cannot do without the mentorship. So because, as you know, in Africa, we don't have many people to mentor young researchers. There are so many people who want to do research in Africa, but there aren't enough people to mentor them. So that's what we've been trying to do together. Currently, we are working with over 30 researchers from over 30 countries in Africa, and working with them day and night, every day of the year, trying to help them to publish papers, trying to help them to do research, graduate with a PhD or graduate with the master's program, learn new courses in statistics. I just finished teaching a course on survival analysis for a group of students in Zimbabwe, and so on. So this workshop is a small selection. It's just a window. It's a small window of the work that we are doing in Africa, just to show what we are doing for a very small. But in actual fact, the work we are doing together covers many people over 30 different countries, and one day we hope to have a bigger workshop that will include all the people that we are working with. I think that will need some big undertaking maybe in the future. We also have with visitors. Like every year, in Manchester, I try to bring in visitors from Africa. Like this year, we had Queensley visiting us for a few weeks. Last year, we had 3 visitors. Every year, we try to bring in visitors from Africa to spend some time in Manchester and try to do more research, because the conditions in Africa, as you know, are not conducive for doing research. They have very limited facilities. The teaching load of lecturers is very high. There are not enough computers. There's constant electricity cut. There's constant internet. Internet speed is slow, and so on and so on. So the conditions are not really good for doing research. But yet, a lot of people I'm working with are producing papers, good quality papers, publishing them in good international journals in spite of all the odds. And we should try to continue to do that. We should try to continue to do that, and more and more in the future. And okay, so anything you like to add, Kinsley? Kinsley, are you Kinsley, are you
Yes,
here?
yeah.
Can you add a few more words to what I have said?
I think you said most of it. The aim of the project and the need to actually encourage a lot of people from the African continent that things like this are possible and they are available and they can key in, especially like visits to Manchester. I know a lot of my friends or scholars, my colleagues that have been saying they want to also, if there's ways they can get research visits. So I mean, we can encourage. We should this work, this part of the work, to encourage a lot of people to have such opportunities, and it's a, I mean, putting it on your CV kinds of boost your CV and opens up other opportunities across other universities globally. So I think this is that's it from me. Yeah.
Okay, that's great. Adam, you want to add more words? Adam, are you there?
I'm there. Well, Prof, I think you and Quincy have said it all. I think I would just also extend our appreciation for your support and constant motivation and encouragement towards the support towards encouraging African researchers and doing all the possible to make sure you support African growth in science and statistics. Likewise to Quinsley and other members who has been in the conference since the first year up to today, which is the third year. We truly appreciate, and hopefully we have more people from Africa. Even here at the conference, I came here and spent, so I met so many Africans. So and I sell this idea to them, and they welcome it. They really appreciate, and they're happy to join the next conference, hopefully. And it's a good thing. We really appreciate your quick intervention because seriously, African are really left behind in terms of support in research. Not that we don't have good people and brilliant people with good ideas, but because of this motivation and availability of resources to do the research. So your presence, it's really inspiring. We appreciate, and I wish everyone best of luck and in his activities and research. Thank you for meeting everyone, and I hope we have a successful and insightful conference. Thank you, everyone, and nice meeting you.
Thank you, Adam. Yeah, as Adam said, like we need to expand our collaborative network. So whoever you know in Africa or outside Africa who are interested in pursuing research that is relevant to Africa, it doesn't need to be Africans. It can be, as I said before, it can be anyone of any color, white, black, yellow, brown, pink, or any color. Anyone who's interested in doing research that will make an impact in Africa. Please network with them. Please connect together. Please let us all work together for the betterment of Africa. And I'm always looking as somebody who's passionate about Africa, about everything to do with Africa. I'm always looking for people, more and more people to work with every day. So the more people we can find, the greater the network, the greater the collaboration, and the greater the impact that we can make to the continent. And one other thing is about visitors. As Kinsley said, I mean, the working conditions in Africa are not conducive for research. So the plan, one of the plans is to bring researchers to Manchester for short periods of time, so that they can do more research while they are here. And for that, we need grants. I mean, the grants for we need especially travel grants for researchers in Africa to come to Europe, and those grants are very limited, as you may know. So if you guys know of any grants that we can apply for, uh, uh, national or international of anything, please let us know, or please share the links with us so that we can apply. We can continue to apply for grants. I mean, I as somebody, I'm always currently will tell you that every month I'm applying for at least 2 grants, or sometimes even more. Unfortunately, not all of them are successful, and but I'm trying. At least I'm trying to apply for grants to bring in researchers from Africa to Manchester for short periods, and sometimes the success rate is very, very small, which is really disheartening. Which is really uh, disappointing. But we should not give up. We should not give up. We should keep trying to apply for more and more grants, and hope that one day we will get a success. That's my philosophy. So you should know of any grants, especially you should know if any grants to bring in researchers from Africa to Europe. Please let us know. Please let us know. Okay, so that's one thing I wanted to emphasize. I think I mean, I've asked many of my colleagues to do the same thing, to look for grants. We can continue to apply for, with the hope that we will succeed one day. All right. I don't have anything more to say. So the first speaker, I'm not sure if the first speaker is around now. The first speaker is Hodo from Somaliland. Are you here, Hodo?
Yeah.
Hi, Hodo. How are you?
Hi, Doctor. I'm good.
Yeah, it's good to see you. So let me introduce Hodo to all of you. Hodo is a young female researcher from Somalia.
Hello.
Hello. Can you mute, please? Can you?
Oh, sorry. Your conference. Do you know what I'm referring to? Prepare slides.
Sorry. Can you mute? Can you? Hodo, can you hear me? Hodo.
Yeah, Doctor. I can, Professor.
All right. Okay. Hodo is a young female researcher from Somaliland. She is extremely talented. She has about 20 papers published in international journals, published or accepted in international journals. I'm trying to get her to Manchester to do a PhD with me. She's an example of a researcher in Africa. Against all the odds, she's trying to do research with very little resources, which is even, I don't know anyone in UK or in Europe or in anywhere, any other part of the world who has done so much research with so little support, with so little resources. So I think I really do admire the work that you're doing, Hodo, in while being in Somaliland, which is one, as you all know, Somaliland is one of the poorest countries in Africa and the world. And in spite of all that, she's an example of a very hardworking person, right? Trying to help her community, not just herself, but her community, by doing research that is relevant for her country. Okay. So thank you, Hodo, for joining this workshop. And I think you can start your presentation now. You will have about 20 minutes for the talk, and followed by 10 minutes of questions. Hello?
Okay. Yeah. I'm here. Thank you, Professor Sarles. for your kind words with me. I'm so welcome, and I would like to share my presentation and then start my presentation. And thank you. I think you see my PowerPoint.
Yes, we can see it, yes.
Okay, thank you very much.
Yes, you can start now. Hold on. Yeah, thank you.
I'm trying to indicate my screen.
Yes.
Yeah. Bismillahirrahmanirrahim, and good afternoon, maybe good morning, some of you here in Somaliland, East Africa. It is afternoon. I'm here. I'm so glad to be the first presenter of this presentation or this workshop for today. And on workshop of Black Heroes of Statistical Research, the third year, I'm Hoda Abdikarim from Somaliland. I'm a researcher. Also, I do researches on reproductive health, specific to maternal and child health. And I'm from Amud University. My title today is Knowledge about Mother to Child Transmission of HIV and its determinants among women of the reproductive age of Somaliland. And I used machine learning analysis. He will do the introduction, the history, objectives, and methodology, the researches and conclusion that will be my content is. To start with the introduction of the dependent variable and the problem I tend to present today. Mother-to-child transmission, which is abbreviated to MTCT of HIV, remains a critical public health challenge, especially in low resources like Somaliland and maybe all in Africa. And while 80%, as the Somaliland Development Health Survey 2020 indicated, while 80% of women in Somaliland are aware of HIV/AIDS, only 55% know that HIV can be transmitted during pregnancy, 60% during delivery, and 64% through breastfeeding. So this history uses machine learning and traditional statistics to identify the key determinants of maternal child transmission knowledge, aiming to inform targeted interventions for future interventions. And also, when we see the comprehensive knowledge of maternal and child transmission, knowledge is defined as these three collectively identifying these three transmission routes: the pregnancy, the delivery, and the breastfeeding. In this research, Somaliland, 84% of the women have comprehensive maternal child transmission knowledge, but only 5% have the comprehensive general HIV knowledge, as the DHS mentioned it. So there's almost geographic, socioeconomic disparities that are significantly influencing the awareness levels, especially between the urban, the rural, and the nomadic populations in Somaliland have nomad populations. Here we have the objectives of the history. The general objective of my history is to model and identify the determinants of maternal and child transmission knowledge among age women in Somaliland by using machine learning models and statistical or traditional statistical methods. The specific objective is my first specific objective was to assess the performance of maternal and child transmission knowledge among reproductive age women in Somaliland, and to evaluate the individual and community level factors, for example, age, education, employment, region, residence, wealth, severity, number of children ever born, age of first marriage, all of these variables that are associated with the mother-to-child transmission knowledge among reproductive age women in Somaliland. The methodology, the history design, the date source, the sample size. The history design, as it is DHS, mostly they use cross-sectional history design as one short time to collect the data for both the dependent variable and independent variables. The cross-sectional history analysis of 2020 Somaliland demographic survey with a sample size of 2,306 women. That's the sample size for this paper for this history. The variables here, we have the outcome variable and the predictors. The outcome variable was the comprehensive maternal child transmission knowledge of which is derived from three different variables, which are the knowledge of mother for HIV transmission during pregnancy, during delivery, and during and breastfeeding. Sorry about that. That last one would be breastfeeding, and then these variables were made into one single variable that was meant to be the dependent variable for this history, and we then made the variable a binary measurement, the categorical measurement of binary yes and no, and the predictive variables we have the age, education, reach, and residence, wealth, media, exposure, HIV knowledge, and other variables like age of first marriage, age of first bears, the an analysis we do here for three analysis, and mostly two traditional statistical methods, binary logistic regression as the measurement of the dependent variable was binary, and machine learning methods that were employed 10 models, for example, random forest, experts, support factor machine, KNN, neural network, the GBM. For performance metrics, the history uses area under curve, sensitivity, specificity, and F1 scores. Here we have the results. The preference and disparities, the first results will be shown here and sharing. The history concluded that 84% of the women have a comprehensive knowledge of maternal to child transmission. The significant regional disparities here, we have the highest regions for knowledge of mothers to child transmission of HIV, Murud and Togder, and the lowest we have Sol and Sinag. The study also showed that nomadic women showed unexpectedly higher knowledge of mother to child transmission of HIV/AIDS. This will be very interesting if we use and show the map of Somaliland. Here we have the regions of Murud Sheikh, Togder, and Sahel that have the highest rates of maternal child transmission knowledge, and the Sol and Senegal regions are having the lowest percentage of maternal to child transmission knowledge. For regression results, based on the objectives of the history, the results is the key predictors after adjustment, the comprehensive HIV knowledge of that shows the more the mothers know about the comprehensive HIV knowledge, it also positively increase the knowledge of mother to child transmission through these three different modes. The region where the knowledge of mother to child transmission was high from Murad-Sheikh, Togder, and Sahel regions, and the nomadic regions that was unexpected as the result of this history. And variables that were not significant after adjustment, wealth, education, and media that were meant to be positively associated or but the history did not show that. The result is in machine learning analysis that we have here employed 10 machine learning analysis, machine learning models And among those 10 machine learning models, which are the random forest, the MP, the probit, logistic regression, GBM, support factor machine, the neural network, DT, which is and expose KNN and random forest. Here, among those 10 machine learning models, random forest stood out the best performing model for this history that achieved an area under care of 0.9%, 95%, that indicated a strong predictive ability. To show more on this specific model that show outperforming the other variables, the other models of machine learning that were employed, the feature selection of this variable here shows that the region was the most significant predictor for the knowledge of mother to child transmission of HIV/AIDS. That was followed closely by other regions, other variables like age, in general, age and age at first marriage, the wealth index, the type of residence, and poverty, education, marital status, mass media exposure, which are derived from the variables of the frequency of watching television and listening radio. For conclusion, they still find the high number of maternal child to transmission knowledge of women, which is 84.9%, below general HIV literacy. Geographic and community factors are stronger predictors than socioeconomic status, since the region and place of residence are strong predictors. And the machine learning models, especially random forest, provide a superior predictive insight compared to other analysis and like traditional statistics. We did the binary logistic regression. For recommendation, they still recommended a targeted educational programs for rural and nomadic regions, since the education of these regions were low compared to the urban regions, and to integrate maternal to child transmission HIV education into antenatal care services and to add the service during pregnancy, and to leverage community networks for health messaging, and to use machine learning for precision public health planning in low-resource settings in Somaliland. For limitations of this history and future directions, limitations here we have, since the data was cross-sectional data, it limits causal inferences, and self-reported knowledge may be subject to bias, since the history was self-reported or DHS were done like that. So, for future work, longitudinal histories to track intervention's impact if there was or if there is intervention, and qualitative research on cultural and stigma barriers, since here there is a stigma barriers and cultural barrier for HIV, and testing mobile health and community-led interventions for both the communities of residences, nomadic and urban, and for the low range regions, Seoul and Seneg. Here for the final message, the machine learning enhances our ability to understand and address health knowledge gaps, in complex low resource sensitivity for impact, since the data-driven complex specific studies can be accelerated progress toward eliminating maternal to child transmission of HIV/AIDS and improving maternal child health in Somaliland and beyond. Oh, it's the end of my presentation. Thank you everyone for your kind support and listening. I think I have done my presentation. I'm ready. Any question?
Thank you, Hoda. Any questions for Hoda?
Yeah, I have one question. I on you one of your results showed that media exposure was not significant after adjustment. What I would love to know maybe what adjustment was made and um, logically, if somebody thinks about it logically, given the internet age, it would be logical to say media exposure or social, whether it's social media or traditional media, is going to impact on the um, result or on the mother to child transmission knowledge or perception. So it's kind of I'm confused how what are the reasons, practical reasons why you think um, media exposure will not impact on that? Because most of these knowledge tools, if you put it on social media, maybe sometimes it goes viral. Even the ones that don't go viral, you still get to get information. So maybe I will need a bit of understanding. Thank you.
Hoda, did you follow the question, Hoda?
Yeah. Hi. Hello. I
Um,
think some people mostly they use social media, but that variable was not in the DHS. If that variable was, I think there might be an impact, but we don't have that kind of variable to analyze. And even here too, for previous time, and the proportion of people who watch TV and radio was too much. And in this area of technology, they use that of the internet and the social media, but that kind of variables were not taken in the DHS, and that's why I think that's where the confusion came.
Okay. Okay, thank you.
Thank you.
any other question for Hoda from the? Mm,
Could I ask a question? If
yes, sure.
I was wondering if how different does this compare to other countries and maybe more specifically sub-Saharan countries or East African countries like Somalia?
In Somalia, we have similar and statistics here, since the data was since the data is the same. Because when the data in Somaliland and Somalia were taken at one time, and you and the WHO and the responsible agencies, they took one time and they are tested, and then we use the data in separate. But if you see in the internet, the data is derived to be or is meant to be the Somalia, but there are different regions, and we use for the analysis those regions. But I can say that the East Africa region is countries like Ethiopia, Kenya, and so on. This history showed much higher compared to other countries like Ethiopia, as I see in my literature.
Thank you
Thank
very
you.
much. Thank you.
Thank you.
Any other questions for Hoda? Hello? Has anyone got more questions? Hoda, has that? I have one question, Hoda. Has this paper been published or is it under review?
No,
Not yet.
I sent to two journalists, but they reject. So I have it now. I will search another journal to submit.
Okay, good. So you use what software did you use for doing the analysis? Is it R software or Python?
R.
R. Okay. Okay, good. Um, good. All right. Any other questions, guys, from the audience? All right. Okay. Once again, thank you very much for your inspiring talk from an inspiring researcher in Somaliland. As I said in the beginning, I mean, you are an inspiration. You are an inspiration for everybody in Africa.
Thank you.
And you're doing so much research in spite of all the difficulties that people have. You know, I must say that in Somaliland, I think one of the facts I read online, whether it's correct or not, I don't know, that about less than 5% of women in Somaliland go to universities. Is that correct? Is that correct, Hodo?
And the last 5 years, the statistics were changed, but the previous it was.
Yeah, it's still a very small number.
Yeah,
It's still a very small number compared to the outside world. So it's a very, very tiny fraction of the population, the female population, go to universities. And in spite of that, you have been a shining example for the other women in Somaliland and not just Somaliland, but the whole of Africa. Yeah. So keep it up. So I hope to see you in Manchester soon. I'm trying.
Thank you, Doctor. Thank you for offering me this garden opportunity. I won't disappoint you.
Okay. Thank you. Okay. Thank you once again. You can. So the next speaker on the schedule is a professor. Anotida he and I, we have something in common. He is currently a professor at the University of British Columbia Math Department. He grew up in Zimbabwe, and part of his first degree he did that. Part of his first degree was at University of Zimbabwe, which was also the case for me. And he went to Oxford to do his PhD there after some time in the US. And thereafter, he became a professor at Sussex University, and now he's a professor at British Columbia, University of British Columbia in Vancouver. So, I know, Tida, I think your scheduled time is supposed to be 11, but are you free now to start? Okay. Thank you.
Thanks, Saralis. Good morning, everyone.
Good morning.
Thanks. Yeah, it's almost 3 a.m. here, so I'm half asleep. So you have to pardon me for that.
Okay. Sorry for disturbing you at this time.
No, not at all. No, not at all. It's my pleasure to be here. Saralis, I didn't realize we have so much in common. See, we have spoken so much about so many things in the past, so it's good that we grew up in the sort of same environment. So I'm delighted to be here.
Yes.
Yeah, I don't know. Shall I start now my talk and
you can take more time if you like, Anotida. So your talk is. I mean, if you need more time, you can take up to 11:20 or something. the UK thing.
No. My question was, can I share now, or maybe
Yeah, yeah, you can.
can I do full view? Maybe I need to.
That's
Uh,
good.
huh? I need to do some full view, but I don't. Maybe here, full screen. What does it go? Yeah, yeah. Um, yeah, it's again. I just wanted to really thank you and Sarales for organizing this, and I know some of you out there. I'm delighted to that you are here. Uh, Joining this workshop. I think it's a very important workshop. It's my first time to be involved in the statistical side of things. My talk is slightly different. As you'll see as I go along, I'm interested in cell dynamics. I'm interested in how single or collective cells migrate in complex environments. They have applications to cancer biology and so on. I'll go through this. There are so many models in the literature about this. Most of them do consider cells as particles, so they really do not look at the cell as an individual, as an entity. And so for this, I that's my focus. That has been my focus for the last maybe 15 years or so, to try to understand both the mechanics and the biochemistry of cells. Yeah, the properties that make up the cells, with the idea that maybe in the future we can make in silico cells that could help with either treatment, cancer treatment, or help with improving maybe wound healing or something like that, inflammation, for example. There are many applications where cancer really plays a key role, and that's what we want to understand, both its mechanics and its biochemistry. So I'll go through this since I have some time, maybe not so gentle, but yeah, I will show both the theory and the numerics and also the experiments. I'll try to go through as much as I can. So I just illustrated to this, understanding how cells migrate through complex environments is crucial to many biological processes, including cancer invasion. I just talked about this, tissue morphogenesis, wound healing, inflammation, and so on. There's quite a lot of literature on this. So cell migration plays a crucial role in many aspects of cell life, cellular life, development, and embryogenesis. Um, in particular, I will show, and this is something that's really nice. In fact, while Ceralis was just talking just now, I was just reading my emails, and there's an email that just came from PLOS Computational Biology, where we submitted this work because we just started working on this very recently, this part of the project. And the editor is sort of asked us to revise, but I think it will be accepted in the long term. So again, the idea is to couple the cytoplasm, the cell cytoplasm, with the nucleus, and we are using geometric surface PDs. And this is a very novel new approach, which I think in many cases most researchers are not able to do. I will show you why, because you're really coupling uh, two manifolds together that are both deforming continuously. And on these manifolds, we have forces that are acting on them. And I will go through this in detail in the next pages. But this is very nice. So the nucleus is the largest and stiffest cellular organ within the cell. Yeah. And it plays a key role of when cells are migrating through confinement. If the nucleus is too stiff, the cell might not be able to go through. And if it is too soft, the cell might be able to go through that confinement. So for cancer invasion, for example, if the nucleus is mutated under some chemical manipulations or concentrations, then this nucleus might become softer. And during migration, cancer invasion, the cells will migrate very fast, very, very fast. If it becomes stiffer, if we mutate it, and the nucleus, the properties of the nucleus becomes stiffer, then the cell will not be able to migrate so fast. So it is really direct impact on cancer. So This is what I'm looking at. I'm not so sure if you can see the movies.
Yes, we
Sir
can.
Alice, do
Yes,
you
we
see them?
can.
Okay. So
Yes.
okay, so I have two examples here. One is a cell, a keratocyte cell that's migrating without so many changes in cell shape, but there are traveling waves that are forming as the cell is moving. You can see immediately the nucleus is the middle, is being dragged along, and there's not so much. There are no confinement, and everything is okay. On this side, you have this dictyostelium cell that's migrating again in a complex environment, but the nucleus is somewhere in the the back end. They did not highlight it here. It's really not playing so much of a role because the nucleus is not so complex. But again, the cell is forming these pseudopods. My focus here is the membrane. The cell membrane is deforming substantially. There are substantial large deformations in the cell membrane. It forms these protrusions that it's sensing the environment, retracts them, it goes left and right, and so on. And it's making decisions on where to go. And this is exactly what we want to reproduce in multi-dimensions, yeah, using modeling. Here's an example of the nucleus, for example, highlighted by Katarina Wolf from Netherlands. So she's one of my collaborators. And all they did was to highlight only the nucleus. And I'm not so sure if you can see some of the nucleus is migrating along these fiber filaments. So these are complex environments. And depending on where they are, some of them are migrating much faster, if you see them, and some of them much slower. So they are going through this dense environment, and they could, depending again on the size of the confinement, on the mechanical properties of the nucleus, the cells might behave differently. Yeah, and so let's look at the modeling that I'm interested in. So I'm interested again in describing mathematically what you're observing here. So what I have is a single cell again expressing these high concentrations of molecular species, actin and myosin. There are two of them. Actin is at the front end and myosin is at the back end. Yeah. When actin is highly concentrated, which is highlighted in green, the cell moves preferentially in that direction. At the back end, which is highlighted in red, you have myosin that's highly concentrated. The myosin helps the cell to contract, and the cell now can move. Yeah, you can see. Yeah, you have green, and this cell moves preferentially. At the back end, you have myosin, which is highly concentrated. Both of them are co-localized. They symmetry breaking. They are not completely uniform everywhere. So there is symmetry breaking, and when you talk of symmetry breaking, you're thinking of pattern formation. Yeah. And so these two molecular species are concentrated on the cell periphery. Yeah. So more or less, I mean, you have some red inside, but they are not really highly concentrated there. So I will focus my modeling only on the cell boundary, only on this evolving, continuously evolving, deforming manifold. I call it surface or manifold, or in 2D, this will be a curve. Yeah. And on this curve, I want to model the molecular species that are living on this continuously deforming surface. So what am I interested in? Two things. I'm interested in mechanics and biochemistry. Yeah. And the mechanics, how do I encode the mechanics? I encode both. The mechanics is really through a geometric evolution law of the cell membrane. And this evolution law allows me to track the cell. So apart from the molecular species which I'm going to solve for, I also do not know the location of the cell boundary. It's an unknown quantity. And I use this geometric evolution. I call it a geometric, it's going to be a PD really, a geometric surface PD for the evolution of the cell membrane. And this same technique, I'm using it here for the cell boundary. I can also use it for the nucleus when I give you an example for the nucleus. So in that case, I'll have two geometric surface PDs which are coupled together. Yeah. So on this, that's all. I have a geometric surface PD that is posed at each material point on the cell membrane. So in the normal direction, I'm just going to go in one direction. I could go tangential, but there's no for me, it's sufficient at the moment to go normal. You can find in the literature a tangential component, but it's mainly used for numerical stability. If you want to equidistribute your mesh, or you want to have a regularized mesh, then the tangential component helps you. Also, to do some analysis, it also can help you. But for my approach, I do not need to equidistribute the mesh using the tangential. Instead, I'm going to use an adaptive strategies that's substantially different. And so I'm just going to deform my cell boundary only in the normal direction. The other things I can handle them in the numerical method. So once I have the PDs themselves, one of the forces will encode the biochemistry, and the biochemistry is for cell polarization, for this symmetry breaking. Yeah, that's the purpose, and it's a surface PD. Yeah, or it could be also bulk if you know what are the bulk species, for example. But at the moment, I'm just going to leave on the surface, so I take a reaction diffusion system with symmetry breaking properties. That's it. Yeah. This is a proof of concept, as you can see. Of course, now I'm working with experimentalists who are deriving. At the moment, I was in France. I just came from France this afternoon, where we are again working on the reaction kinetics that we want to get in these reaction diffusion equations based from chemistry, from experiments. Sorry, and so. But for today, I'm just going to be hypothetical in terms of these kinetics. So very, very quickly, I might, Serales, you might give me a thumb up when I've 10 minutes or so, because I always run out, because there's quite a lot. I'm very fascinated about this work, so I tend to describe quite a lot, but the basics are, we have this reaction diffusion equation as it's posed on an evolving gamma of T. Yeah. This is the material velocity here in terms of, if you think of fluid dynamics, because everything is deforming here, I can write it in terms of this rate of the material velocity of B, rate of change of B, plus the velocity dotted with the gradient of B. So this is the convective part, yeah, describing evolution of the membrane. So everything is being transported by the velocity, material velocity V. Now I add a second term, and this second term is plus B. B is the species. So the B's, they are being either depleted in concentration, or there is growth in concentration by this divergence of material V. And also, in fluid dynamics, this also says whether your material is compressible or incompressible. So you have the divergence of V. And that was, you can also call upon that theory. And that's this term, basically. Again, it's involved, it's describing how the surface gamma of T is behaving in the presence of the material velocity and in the presence of the B species. Now, the B species, I assume them that they move randomly on the surface. So I have surface diffusion, and when I have surface diffusion, I have the Laplace-Beltrami operator, the divergence of the gradient of B. dB is the diagonal metric of the diffusion coefficients. F of B here is the reaction kinetics. As I said, this normally should come from experiments. This is the only place where the B species, I take n of them are interacting. So this is known as the reaction kinetics. Yeah, and this is very important. It should be non-linear, and should give rise to symmetry breaking, to pattern formation. Classical sort of conditions that are required for pattern formation. Yeah. What else? Yeah, the V is the important one. So if there's no growth, if gamma of T was just stationary, this term will vanish. The divergence of V will be 0. The V here will be 0. So you just have exactly the rate of change of B with respect to time T, which has been studied by Turin since 1952, and there's lots of work on that on stationary surfaces. Well, domains, not surfaces as such, but planar domains are spatial surfaces. Yeah. And so what is the difference? Now, is this material velocity? This is a very important one. Yeah. This material velocity, we're going to model it either from experiments, if you know, if you're given experimental observations, or we are just going to say what we have, and this is true from from this movie, the cell membrane is energetic. It's energetic cell membrane, so it has surface tension in it. It has bending energy, for example. So you can call upon the Wilmot flow sort of framework and write down what is this V. I write it in this format. This is through a force balance equation. If you take this is the viscous type, this is the mechanics actually without inertia. I just take this type. If you take it to the other side, you have zero. So it's a force balance equation. So this the viscous force is being balanced with the rest of the forces. The first one, I mean, generally I write it as a G function because it's a complex function that encodes all the forces. Polarization for pseudopods formation when the cell breaks symmetry through this B species, and that's what I'm describing here. You have retraction, the cell is retracting, and after, when it forms the pseudopods, it retracts. If it doesn't want to go in that direction, it forms another one, and so on. And then the bending energy, the cell is restricting too much bending. So, and it is described by this Wilmot flow. I should have said, actually, this is the surface tension as well. You can have it in terms of the mean curvature. Here, you have volume constraint, which is associated with the Lagrange multiplier lambda t. Again, we have to have a model for that. Normally, it's a differential equation. But it depends whether you want conservation or you want constraint. You have internal forces, which, I mean, there are lots of examples of how external forces are exerted on the cell membrane through microtubules, through intermediate filaments, and so on. Yeah, external forces. Cells like you and me, when they walk, they don't want to bump into another human being or another cell. So they have to respect each other. If there's an object, if there's an obstacle, they cannot go through. They have to squeeze in if they want to go through. So there is that interaction between the cell and the environment, and the filament and the filaments of the extracellular matrix, which are collagen filaments, for example. But also, you might have cell-to-cell interaction. Cells themselves, talking to each other. But also, cells can sense chemicals. These are forces exerted. They can sense the force, a large force, and they can migrate preferentially in that direction. Or they adhere. They adhere to the substrate, either in 2D or in 3D, depending on where you are. And so you have adhesive forces. You can encode this. So, I mean, this is a very general framework that I'm proposing here. It depends really on the biology. But the underlying forces, the key forces for me, are the surface tension here, which is given in terms of the mean curvature H, the bending energy, again, given in terms of the mean curvature H. It's non-linear because you have H cubed. Also, you have the Laplace spectrum of H, where H is itself the Laplace spectrum, second order of the material points S, which lives on gamma, the material point. So I take the Laplace spectrum, of the Laplace of the material points, and then I have a fourth order. This is a fourth order PD in S. So when you compute this PD, it's really fourth order, and the rest of them are forces. And I keep the lambda t, and I change the forces depending on the biology. The rest of either internal or external, or if I don't have polarization, for example, cells migrating through microfluidic devices, there are no chemical species that we are looking at. It's just the constraint. It's a constrained environment, and therefore we don't need the polarization, the Turing model. As I said, I'm moving in the normal direction. Okay, I've said enough already. So let me give you some examples. I use finite elements. I'm going to be very blunt. I just use evolving surface finite elements. If you want the details, I can give you this. We can solve the model in 3D. Here I'm using Maynard's reaction kinetics. They are well established since 1972. And so I just use this for polarization. They are well known that they do give rise to symmetry breaking. And so as an example, I have this cell that's migrating in a complex environment. As you can see, it's a neutrophil cell that's chasing bacteria in the presence of white blood cells. And it pushes these white blood cells out of the way because it wants to feed, and the bacteria is trying to survive as much as it can. I mean, it's going through all these pores, and finally it's engulfed by the cell. So what has happened is the cell is migrating in a complex environment. The environment is made out of deformable objects. These are white blood cells. And so, can the mathematics reproduce this experiment? In the absence of the bacteria, I could have put the bacteria as well. I think this would be very nice to do. I can do that maybe in the future. And I would not just need to put a chemotrectant so it can follow. And the speed of the cell is much, much higher than the speed of the chemotrectant, the speed of the bacteria. I think I am just thinking on top of my head. Actually, I might put the bacteria in the next simulation. And yeah, as you can see, the framework really does describe what you're observing. And really the question is, can we do more? How can we do much more when we have to cell interactions, for example, in the presence of a chemotrectant? So I have 10 cells. On these 10 cells, there are 10 geometric surface PDs, one on each cell. And these PDs, within this boundary, within this environment, they're talking to each other. The cells respect each other. They can bump into each other, they can squeeze to each other, but they really do not overlap as much. And so this is exactly what you observe biologically. And one could use this as a starting point for the formation of tissue, for example, of vasculature, whatever. This is really the idea. I will just go and try and give you, because I don't have much time, I just realized now, I want to give you an example again, biological example. This is work we just submitted for publication as well. In fact, this is the PLOS biology work that has just been reviewed. So I have a nucleus and I have this cell cytoplasm, and the cell is being translocated. It's translocating. It's not migrating as such. It's being pushed through a microchannel by a pressure. So they have a pressure gradient that pushes the cell across, and everything is constant in terms of the mechanics of the cell, in terms of, there's no biochemistry here, but everything is constant except the width of the channel. So we change the width to see whether, if the width is very small, tiny one, does the cell go through, what would be the optimal width that the cell can go through. All these we can quantify. But as I said, I don't have enough time to go through all the details, all the sort of outcomes of this work. But I'll show you just some of the main results. Yeah. Okay. Let's go through the model. I'm really not going to go through it again. Maybe I should just say one more thing. I didn't talk about this Lagrange multiplier. This is for the membrane. It's given by a differential equation. And this differential equation, you can show that if A is the area, the reference area, the Lagrange multiplier oscillates around this reference area. It does not decay to zero. It does not expand. The Lagrange multiplier does not explode to infinity. So it's well bounded and it's positive. And that's what you want. Not necessarily positive, sorry. It's a Lagrange multiplier. It doesn't go to zero, either negatively or positively. Yeah. And so I have this the viscous, the volume constraint, I have the surface tension, I have the bending energy, and I have other forces. In particular, I want to highlight this mechanical force. So I can pose mechanics on the cell membrane. I take two approaches. One is a linear elasticity. The other one is viscoelastic, because I don't know which mechanics are better, and I try to model these two together. And then I have external forces, which is the barrier. The barrier for the cytoplasm, but also the nucleus. Okay, the cell plasma membrane has to know where the nucleus is. The nucleus itself must know where the cell membrane is. They are interacting. You cannot have a nucleus that's migrating much faster than the cytoplasm and it will go out. That's biologically unrealistic. Or you can have the plasma membrane deforming substantially, that the nucleus is going to be left behind outside. This cannot be. So you have to model these forces. And I will not go into the details, but if you are interested, some of these details, for example, here. You don't want the membrane itself to self-intersect. we want to avoid that. So we design these forces and we we impose them on the cell membrane. This is the mechanics, and this is the first one. I think the elastic one, it's given here, and the second one, the viscoelastic, it's given below here. But I'm not going to go into the details simply because I don't have the time. What I was going to say is the force, the mechanical force which is given here. All you need is to know the stress, the sigma of u. I mean, I chose linear elasticity, but someone can choose something else, and it's projected in the normal direction of the cell membrane. The C here is for cell membrane. And the forces, again, external forces, there are so many of them, as I just described. Interactions between cell and the external physical constraint, that the barrier, for example. I have another one, the active pressure, because I said the cell is being pushed by the pressure gradient. So is there. I also have nucleus and cytoplasm interaction forces. Okay, and so on. So I can now write the geometric surface PDEs, both of them, for the cell plasma membrane and the nucleus. But they look very similar. Of course, they have properties that are very similar, but the mechanical, the model parameters are different. That's one thing. The other thing is the constraint, the forces. Some of them I have here. I mean, self repulsion, which is okay. I have mechanics, but different properties. I have external forces, but they will be different depending on the manifold. And for example, here for the cell, I have the barrier, I have the pressure term, I have the repulsion, for example, and a dragging force to drag the nucleus with it. Yeah. While the nucleus, it does not. It is a drag. This is direct. This is related to the nucleus. The dr I have here, but you also have a repulsion. You have a barrier, but not more. For example, it doesn't have the pressure. The pressure is being exerted from outside onto the cell plasma membrane, not the nucleus itself. And so you can have different forces. Everything is projected in the normal direction. Okay, I hope I've convinced you that these are the two right models. I really have to again highlight this. These two PDEs interact through this DFD and also through these DR forces. Yeah, the dragging force and also the D. What I have it here. Where is that? In fact, it's the wrong one. Sorry, there's a typo here. It's the elastic one. It should be the elastic one. So this is a typo, really. The interaction between the cell and the nucleus using an elastic force. So they are connected by an elastic force. And so this should be EL, really. There's a typo here. See, I need to write that down and make the correction. Okay, so yeah, that's it. So I take you down to some examples. Maybe this is I hope you can appreciate. It's the same cell here that's migrating in complex environment with fibers. You have the nucleus. I will look here at the nucleus in green, and it's the pseudopods are forming, but you have this very long philopodia. Philopodium that is forming. It's exploring, and the cell is following. The rest of the cell is forming. There's another cell that's coming up behind it. You can see almost immediately, and it's philopodia. It's much smaller. Why? Because the cell is being guided by the cell that's in front of it. It does not need to form as much the longer philopodia because it's sensing itself. There is what they call the lead cell migration. So the leading cell is leading, and the other cells are following. Follow the leader, as they call it. So we just want to see whether our numerics here. I did not even model many cells. I'm just looking at the formation of the philopodia in the presence of this complex environment. In this case, we allow it to retract. We are really not migrating that much. So this again is a proof of concept that this philopodia can form under this geometric surface PDE just for the cytoplasm, because the nucleus is not forming philopodia. I mean, it's clear. It's just being dragged along. So we did not model the nucleus here. But we can, I'll show you in a minute where we model the nucleus. So this was just one example. Again, the formation of philopodia. Here, I will look just at the nucleus. So we tested this for just for the cell, if you just model the cell plasma membrane, or if you just model the nucleus by itself. So in this experiment, what they showed, and this is the work by Kao and Ahmad. I call him Ahmad. He's a UCL, a friend of mine, and so he gave me this work and said, okay, the cell in green is migrating through these complex environments of different sizes, and the nucleus is the one that's really holding back the cell, depending on its stiffness. You can see in green, the cytoplasm is free. It's like a gel. It goes through very quickly through this. The confinement does not hinder its deformation at all. It is the nucleus, and the nucleus now, through this confined space between these two big objects, it's struggling to go through. It takes about 42 minutes to make even halfway through, and then it takes about 2 more minutes to go through. Once the bulk of it has sense that it can go through, then it just goes through very, very quickly. And now we just looking at the nucleus. This is our computational model. Here, the mechanics of the cell are much stiffer, and here the mechanics are much, much softer. You can see sometimes the cell can not go through, and sometimes it can go through. Again, this is just as a proof of concept. We need more biology. And so I'll be talking to Ahmad about this. As I said, we just developed this in the last three months, all this framework since May. So I recruited this postdoc in France in May, and she has done extremely well since that time. Now I want to look at cell migration in confinement. I described to you about this pressure that's being applied to a cell as it is going through. Think of a pen. Think of a pen that you want the ink to go the other side, but really what you want is to push the cell to go through this really confinement with a very tight.
You have 2 minutes left. Oh, you have 1 minute left. Sorry to pressure you.
Exactly.
Yeah, sorry.
Yeah, because I will need some. Yeah, but yeah, exactly. So we want to understand this. We can apply, and just very quickly, I'll show you that it can. You have two manifolds that are interacting, and this is important. And the cell, can go through, depending on the thickness of this of this microchannel. And if you make the thickness much smaller, the cell really struggles to go. It takes a much longer time to go. And we can also look at cell-to-cell interactions if you wanted to extend this to this complex. And I think I will stop here. I will skip these ones. I wanted to show you other sort of results, but what I'll just say is I've developed this geometric surface PD approach for modeling single cell migration. Collective cell migration is a byproduct of this. We can model it as well. Yeah. There's quite a lot of challenging here. I didn't show you any mathematical or numerical analysis, how I solve them and so on. The coupling of mechanics with biochemistry, this is really, we're just at the tip of the iceberg. Really, we are starting this so much that we don't know. Yeah, I didn't model intercellular devices. I didn't model any much more complex environments than I just showed you here. So these are my collaborators, in particular in Nice, but I also have PhD and master students here. Here at UBC who are working also on this type of problems, and also my other postdoc in Italy. I have my collaborators from Sussex, Chandra Shaka, who has also contributed substantially to some of this methodology, and Laura Matthew, a former PhD student. Okay, I think that's it.
Thank you.
Just acknowledging my funding. Yeah, I stop here.
Thank you very much, Anotida. You are such an inspiration for a lot of us. I think one of the things I admire you for is the amount of funding that you get. You are so successful in getting funding. We on the other hand, as I said, I don't think you were in the beginning of the workshop. One of the things we are trying to do is to bring in researchers from Africa on short visits to the UK. And I've been able to get some funds, but they're extremely difficult to get. They're becoming more and more difficult to get. do you have advice with the success you have? I mean, which is enormous. Do you have ideas about like what kind of grants we can apply for with respect to bringing researchers from Africa for short periods, like 1 or 2 months?
Yeah, I have some. My term on the London Mathematical Society board is coming to an end. They do have funding.
Yeah, I have.
Yeah, for the mentoring for the mom. You might know about mom.
Yes, I do. I have a
Okay, that's one. Although they really do not in the past, because I had a mom grant 2010, I think it was, to 2012. You could cost for people to visit, but it's so expensive to visit. So it's better to use the money for the researcher to go to Africa. Sometimes it's cheaper, but it depends. But you can use the money as you wish, and just fine. But I think there are. It's funding is really a big issue. I mean,
Yeah,
this is uh, one of the things we are trying to do is also to getting funding from here, Canada. But if you once you do that, then people have to come to Canada. If you are looking at the UK, it has to be UK funding. It's very difficult. I would maybe in the future what you could potentially do is to lobby the EPSRC. I was thinking of doing that while I was in the UK. No, go and look at the board of directors and lobby them about funding for such things. The ICMS is one of them. The Newton Isaac Newton Institute is another one, but they are funded by EPSRC. So if you could go and talk to ICMS, talk to the Isaac Newton Institute directors, and apply for money, because they are funded by EPSRC, and apply for funding to EPSRC, I think you have better chances of getting them. And using the Newton Institute, the ICMS, as hosting, because what you want is to bring the researchers, but also to have a broader impact beyond just visiting these institutes. So they can visit other universities. If you can, I mean, I am happy to give you. This is just from the top of my head at the moment. You can, I can give you some ideas, but you can talk to the directors, have lobby for them, and try and say, okay, we want to apply to the EPRC with this grant. So much money will be managed through Isaac Newton Institute or the ICMS, and you can bring researchers for certain periods of time. I think this is doable. This is actually it's doable,
yeah,
because at the end of the day, for EPSRC, you have to apply for money. You apply for funding,
yeah,
I mean, ideas are ideas. If you bring the ideas, if they say, "Oh, this is rubbish," that's fine. But if you don't apply, then they will never say this is rubbish.
yeah,
I
of
think
course.
one should just. I mean, you say I get lots of funding. Because I just close my eyes and say, I apply. This is the British Columbia Foundation of Non-Animal. It's not well known. Mathematicians do not apply to these grants at all. I just applied, and they really
Yeah,
liked the idea, and they said, why not? So you just have to be innovative how you look at funding bodies. So that would be my Sarales. That's my suggestion. I don't want to run over time, but yeah,
yeah.
we can
Thank
have
you.
a discussion about this.
Later on, yeah. Thank you.
Yeah.
Any other question?
Yes, I
Vincent?
have a question. Yeah. Thank you so much, Anatude, for this very interesting. In fact, I was just fixated on the idea, the process, the innovative aspect of the work. Quite really, really interesting. And I wanted
Thank you.
to,
Yes.
yeah, I wanted to, like the composition of the cell is fixed, even though it deforms. The boundary is flexible, but the composition, not the movement, because it's like a volume, depending on how, the cup, or depending on the shape of the boundary, to take that shape. But its composition is fixed because all the cell makeup is still there. when it comes out, it goes into a different shape. If it's trying to go through a complex environment, it kind of squeezes. So did the model take into account the composition of the cell while modeling it?
Excellent. So you're absolutely right. I mean, I've been studying the cell for the last 15 years when I started looking at this. The cell is made out of so much material inside. Apart from the nucleus, you have intermediate filament, you have keratin material, you have depends on the cell. So all I'm doing here is I'm ignoring all that. I'm putting it in the Lagrange multiplier lambda. Okay. So the Lagrange multiplier monitors the deformation, the cytosolic deformation, the interior of the cell. So it's never constant. I could plot the lambda, it always varies because when it's constrained or the perimeter, you can see, I forgot to talk about this. We put also a constraint on the perimeter that the perimeter cannot go forever. So these things are important, but it means that your interior, the composition is changing in time. It's never constant. The cell loses its water through osmosis, for example. And when you squeeze the nucleus, it also ruptures. And there's evidence in the literature that shows this, and its volume shrinks. But the cell can repair. I haven't modeled this, but that Lagrange multiplier encodes this loss of volume or gain of volume, gain in composition or loss in composition. I hope that answers.
Yeah, it does. Thank you. One more question, please. When the cell was migrating, you have the green part and the red part. So you said the green indicates it's more active because at that green, it kind of follows the direction of that green is what determines the path. Yeah.
Right. Yeah.
And that has to do with like the material velocity. Is that what has to do with the material velocity?
No. Okay. I did not explain this very well. So the green, think of the molecular. So I talk about acting as a concentration. So when acting is uniformly concentrated around the cell periphery, the cell cannot make a decision.
Okay.
Because the concentration everywhere is the same. Now, when the concentration of the actin becomes concentrated on one end, which is what you saw, when it's highly and everywhere else it's almost non-existent, then the cell really deforms in that direction.
Okay.
So it's concentration driven.
Okay. Oh, okay.
Okay. So the material velocity in that part of the membrane has high concentrations.
All right. Okay.
So the velocity is driven by the molecular species.
Okay.
Yeah.
This alignment of cells, when you looked at two cells, and the first one was moving, when the second one came, it kind of aligned with the first one.
Yeah.
It's like follow the leader. You say follow the leader.
Yeah.
Mental. Yeah. So when that means cells are aligning. The alignment of cells in migration mode, it follows like a swarm. Like fishes when they move, they just
super.
Yeah, the one with the highest environment or force tension, the others just follow. So that's the way the cells do as well.
Very, very. So when you have a cancer, you have a tumor. Yeah, you have a tumor. What is known as angiogenesis is when the cancer is running out of food, it sends cells to secondary sites. It wants to send the individual cells to somewhere. It's like an army. It's like an army. Okay, I'm going to die somehow. I really need to send my army somewhere to start another settlement somewhere. And it sends these cells. The first cell lives by itself, but immediately the next cell is following it. As you have described, it's like fish. Once one starts to go, the next one comes, and immediately a lot of them come. They go. They are just following each other. Exactly.
Okay, thank you so much.
Yeah. Yeah, but I would love to model fish swimming. I think that's we can. There's no reason why we cannot use this approach. But I don't have the data. But it's a very good example.
Okay, thank you.
Yeah.
Thank you, Queensley. Thank you, Anatida. I think, yeah, I think your talk has been really inspirational. I think we can talk another time about the grants thing, Anatida.
Absolutely.
So, because that's something we need to work on really hard, especially we here in the UK, because I mean the grants I've been getting are really small ones from LMS and other places, but we need to try something bigger.
That's right.
I think your help and expertise will be much appreciated. All right.
No worries. I'm happy to discuss.
Thank you.
Yeah.
Thanks
And
again.
Queensley, nice to hear from you. Yeah. Yeah.
Thank you. So the next talk is by our own fantastic lady, superwoman, Queensley. So Queensley is gonna be the next. Is gonna provide the next talk. It's gonna be on climate change, STEM, and disability.
Okay, thank you. Can you see my slides?
Yes, of course.
All right. I'll be presenting on climate change, STEM, and disability, and drawing out lessons from a collection of impact-driven projects that we've done that empower marginalized communities. So I'll be providing the background, the aims and objective of the presentation. I'll highlight 4 case studies and provide limitations and conclusion. We know that globally, female scientists have actually constitute less than 30% of researchers, according to UNESCO, and even the representation of African women are way lower. But when you look at the African Union Agenda 2063, they are so keen, they emphasize in the Aspiration 6 that women should and have to be an integral part of the continent's development. I mean, more so that women make up close to 50% or slightly around that. But there's very little known about the statistics of women in mathematics, institutional policy structures that surround or support women in mathematics, women in STEM. So there's very poor visibility on that. As well, there are STEM initiatives that center around persons with disabilities are very few, especially those that bring in persons with disabilities as co-creators, not just only as beneficiaries. And this also applies in the aspect of climate initiatives. So the aim of this is to showcase practical impact-centered STEM interventions which expand the opportunity for marginalized groups in both in Nigeria and across Africa. I would love to present the lessons I've learned, insights, evidences from the portfolio. These projects are meant to shed more light on how rigorous data, participatory design, and cross-sector partnerships can produce potential and scalable solutions in the area of education, climate, resilience, and inclusive innovation. Another aim of this is to help present simple, clear models that others can replicate for community engagement, for monitoring and evaluation, for getting little funds that different within the 4 different African contexts. So the first project is I am a STEM advocate. We ran this project in 2024-2025, and it was funded by Alumni Project funding from Germany. I was the leader. I led the project with, I collaborated with Dr. Chiwendu Maduweze and Christina Oyewole. The aim was to raise awareness, dismantle the misconceptions of STEM, and foster supportive environment for girls to engage with STEM subjects. So it was done for Nigeria. It was done in Nigeria. We reached across 3 states in the north, North Central, South, and then the Southwest of the country, reaching over 2,500 girls. And these were girls in senior secondary school. So we're looking at SS1 and SS2 students. So most of the activities were about science subjects, about financial literacy, games, how we can inculcate games in understanding science subjects, coding, emotional intelligence, how you can recognize facial patterns, facial expressions, and all that. And as well, there was also, I mean, that was not in the plan, but from the outcome of the project, we also had career counseling. Some of the measurable outcomes were real enrollment in further maths. So that's what you call advanced maths in senior secondary school. So we had a lady who was a kind of leader, maybe like a prefect or so in the school. She had told many of her mates to drop further maths because it was useless to them going forward. But when we came and kind of showed the usefulness of that, she had to come out and start advocating for it again, and even took up the cause. So we had, I noticed that a lot of students, female, and it was female students, a lot of female students were not even so, they were much more interested in the financial literacy and how they can set goals for their progress, for their career, for their lives. It was quite interesting to see that after maybe hold for the whole day, up to 5 hours, because of the funding, we had to kind of squeeze everything within. Girls will still wait for 1 to 2 hours, just wanting to discuss about their future, about their personal problems. So it was really beautiful to see that. One of the schools we did this advocacy project, and so just one of the schools I kind of ran a fast exploratory analysis to see their take on overall interest in pursuing STEM after the program. And as you can see, the before is represented by the blue, and the after is represented by the orange. And we see that there's a substantial positive shift in the students' mindset in favor of STEM. And this is quite significant because about 70% of the girls we met they've never heard of STEM before. They've never heard of the word STEM. They don't know about it. It was really shocking to me. Like one school, you will have about 2,000 girls, and that's just SS1 and SS2 students. Another school, you will have about 700 girls, something like that. So it was fulfilling project. But when we broke it down to see different, this one might be so small, to see different disaggregated data, to see their take in terms of like confidence in STEM subjects, knowledge in STEM careers, interest in STEM subjects like that, the response actually varied. And this is just for girls in a school in Ibadan. That we ran, because there's so much data, but we have to take our time to actually analyze all that data. The second project is actually a disability inclusive climate action. This took place between 2024 and 2025. And it was funded by Disability Rights Fund. I led this project with the Center for Citizens with Disabilities. And we took a baseline survey to understand the climate impact on persons with disabilities and their livelihoods. There were about 104 of them, and that was specifically in Abia State in Southeast Nigeria. The stories from the persons with disabilities included how the climate change has really impacted on their lives. For example, a good number of them are farmers, and some of them said, especially when there's flood, they can't protect their farms. A lot of thieves go in and steal their harvest, especially when it's close to harvest time and the crops are there, they will steal it. And so this got me thinking about what about designing a greenhouse for persons with disabilities in this state that is housed within maybe other value-adding initiatives like food processing, energy, renewable energy, and all that. So that was the idea that started growing in my mind. And the data analysis actually showed that there was a more positive response, relatively, from the southern part of the state than from the northern part of the state, because we looked at the three senatorial districts: Abia South, Abia Central, Abia North. And when I started noticing what the data was saying, I decided to go and check why is there this trend. And it boiled down to the fact that in Abia South, there's a commercial center. It's a commercial center. We have the Aba Commercial Center there. And it made sense to me that the environment itself will be quite supportive in terms of economic income and protection, maybe like associations and all that. So some of the measurable outcomes were that during the validation session, it was quite beautiful to see a lot of interest in the project, especially from the Ministry of Environment, Ministry of Budget and Planning, Ministry of Health. So we had a lot of representatives from there. And during that time, that was when also the Abia State government launched a persons with disability data capture, because we noticed that there was no major database for persons with disabilities. Even across Nigeria, there's no comprehensive database. So that is really a problem, because when you don't have the data, you can't even work. And then the Abia State Ministry of Environment actually established a disability inclusive climate DEX. Further, given the rigor of the work, the quality, we were able to I was able to establish links with Harvard Law School Project on Disability, and that brought about two publications. One was a UNICEF toolkit, which actually focused on youth with disability, and that's going across the world to see how they are impacted, and we are the ones that took care of the Nigerian portion. And there's also a paper on how the perceptions of Abia State persons with disabilities in regards to climate change and the different policy recommendations that can be undertaken. It's been published in sustainability. Then the third project is capacity building for women in mathematics. And that is, this took place in 2023, 2024. There was one in 2023 and one in 2024, and was funded by the Committee for Women in Mathematics, International Mathematical Union, under the International Mathematical Union. It was led by, the first one was led by Dr. Chiwendu Madube, and the second one was led by Dr. Mwawi. So I was a collaborator in this. The first one dealt with looking at how to break barriers of girls through networking, and the second one actually focused on work-life balance for African women. Cumulatively, we reached across to 250 women plus. That's more than 250 women across Africa, across 15 countries. There were over 15 countries. And a major aspect of it was mentorship, but there was also research, especially for from the second project. And I led the sub research on mathematics and maternity, because that's really very dear to me as a mother as well in mathematics. So we have ongoing, we're still working on the papers, about 5 technical papers on childcare, mentoring, and now we have a collection of data that focuses on institutional and policy structures, which has not been available. Then there was a very interesting aspect where we organized mathematical poetry, which I led. A lot of people responded, especially girls from secondary school, and how mathematics can promote the joy, how poetry can promote the joy of mathematics, how it can motivate. We had 44 entries from different African countries, and the winner was a lady called Tia Mike. She was from Ghana, or she's from Ghana. And I'll just read the first verse of her poem. It's called Beauty of a Lady with Numbers. In the realm where numbers dance and equations sing, there dwells a lady with passion in her eyes that gleam. Let me look at it. Her heart, a sanctuary for the language of math, where patterns unfold and mysteries untangle their path. So I'm also into music. So it got me thinking about how to infuse a lot of music and art into mathematics, especially for young girls and boys as well, anyways, to help give them that joy, to help make the pathway not to look so straight, so flexible, so inflexible, because most times they say mathematics is one way, but I believe it's supposed to be quite flexible. Yeah. And it also got me thinking about the STEM gender gap, especially when we looked at the policy and the institutional aspects, structures. I started looking at theoretical and conceptual studies that are existing. And I found out that there are single theories that have tried to explain this STEM gender gap, like scaling that apply liberal feminist theory. And we have Kim that adopted the critical race theory. And we also have the STEM belonging. Then, from the work and from the experience, I know that it's multiple perspectives or multiple aspects that actually bring about this, because there is not, people are aware that there's a STEM gender gap, but it still persists despite the awareness. So there are very few applications of multiple theories. We've had those who have synthesized divergent theoretical perspectives and those who have compared theoretical and empirical concepts. But the one that caught my attention was Miner, who compared the individual and social structural constructs. And Dev also improved on this Miner's work by looking at the cultural aspect, the cultural beliefs. I decided to improve on that structure. So by constructing a new conceptual model. And the aim of doing this is to improve on the current state of STEM education management system, because it is critical. I found out that one of the reasons for the persistence has to do with this education management aspect, the risk management aspect of STEM. So I improved on the Miner's model by combining both individual level and system level perspectives. So while Miner was looking at them from a siloed angle, my idea was to combine them and use the support of equity and social justice theory to actually support the construction. And this will help to explain not only whether gender gaps exist, but why they persist and what institutional levels can disrupt them. And so this is the construction where we have the institutional structures, control, where it goes from institutional justice features. So I'm saying that the institutional structures control the environmental factors which shape the input and outcomes that are available to women. Now, this also further determines the objective outcome and input ratios, leading now to the subjective perception of equity. Now, this is at the level of individual. Once that perception is that of injustice, the individual's behavior towards their institution can now does degenerate, contributing to that gender gap. And these gaps now feed back into the system. So we see a feedback loop, a feedback into the system, affecting recognition where the women are not recognized, representation, they are not represented at the table, which further compounds the distortion and the bias in institutional policies and individual perceptions. So I and Dr. Dr. Chiwendu have submitted this paper for peer review. The last project is STEM and disability. So the first two projects actually got me deeply pondering about how persons with disabilities actually fare in the STEM field. And this gave birth to this fourth project funded by the Better World Grant, University of Manchester, collaborating with Professor Sarah Liz Naduraja and Dr. Christiana Charalambos. The aim is to provide disability-centered, low-cost tech products, and there are two teams. We have one in Manchester and the other one in Jomo Kenyatta University. The main activities are mentoring and developing the simple prototypes, and most of the prototypes are about 70 to 80% completed. I also had an increased understanding of the policy and institutional dynamics that surround disability inclusive perspectives within the STEM field. We have the team from Manchester is looking at Chipper. Chipper is just a very small device that is imputed with a chip, and it helps because the problem is that the communication system within the STEM field is quite taxing to persons with disabilities. I mean, their policies, the documentation and all that. So it's this chip is meant express themselves better. Like, I do not understand this place, so they can press, and it will be color coded, so they can press the particular color, and they can get an explanation that targets that aspect. And for the team in Jomo Kenyatta University, it's an inclusive AI assistant for blind STEM students, a wearable device that is powered by AI. We have a website, so please visit our website. Thank you. So the limitations of these case studies are the sustainability aspect of the projects. Most projects are not sustainable, and why? Because they're usually at the mercy of funding continuity, and this process shows that there are very huge gaps that exist on African women in STEM, and even disability, that which is worse. The social media presence is almost non-existent for women in STEM, although we have women in STEM, but the projects that come out of it, the impact, the whole 9 yards, there's very little visibility. Scaling the prototypes is a key aspect of sustainability, but that also depends on funding and institutional buy-in. We also have the cultural barriers and limited mitigation tactics. But overall, this got me thinking about, even if I don't get a grant, how can I use from my own pocket, do little pet projects that can help my community, and things like give an award for the best graduating student in the secondary school, maybe best female mathematician or STEM female students, things like that, or promoting tertiary level students, micro businesses. Some of them have so many little businesses that they do. So those are the things I'm thinking to work into 2026 and at least budget that from my own pocket to be able to help my environment. So in conclusion, I would say that there's enough zeal and willing ground soldiers on the African continent. There is enough zeal to actually want to learn, but direction is a problem, which has to do with mentoring, and funding is quite scarce, as Arelise has actually emphasized a lot on that. But I noticed that even very small grants go a very long way, actually. Especially if it is if the work is being done here in Africa or within communities here. So the call to action is to partner, to fund wherever you are across the continent, to pilot, to adopt, to mentor. Because where we have many of these little projects springing up in different areas of the continent, collectively will make a big difference. So these are the references, and thank you very much for listening. I appreciate it.
Thank you, Quincy. It's a beautiful, inspiring talk. I think we need to do more STEM projects in Africa, and if I can be part of any of those projects, please keep me involved. I'm happy to be involved. I mean, I think there's a much greater need for STEM activities in Africa, and we'll try our best to find funding. We will. Yeah. Thank you again. Any other questions from the audience for Kingsley? Hello.
Saralis.
Yes.
Yeah, do you hear me?
Yes, of course.
Yeah. I just wanted to say also very, very nice talk, Queensley. I think this is a very nice story that you could I think it deserves to be told in some fashion somehow. I don't know. I mean, I'm thinking. I wrote in the chat about the poem. You could maybe approach something like the Royal Society. There some competition for the best pictures of the year or something like that. But I think if you have a picture with that poem, I think that could also be very nice. It's a for the student. I think that would be really a good inspiration. But you can look at some newsletters where you can write that, where she can send and write, and have that exposure. Maybe I think that would be very, very helpful. So thanks for bringing that. Very, very nice.
Thank you.
Yeah. I think curiously we can try there's something called the IMA newsletter, which comes out, I don't know, every 2-3 months. I think we can try that. We can try that and see if they're willing to publish that or what. Yeah. And any other comments for Kingsley's talk?
I was wondering whether whether you have some insights in terms of you are talking about, is it over 15 countries where you are studying these gender gaps? And I'm wondering whether there is a kind of pattern that comes out over those 15 countries, and also probably maybe in terms of initiatives, just some highlights on initiatives that seem to narrow those gender gaps.
Okay, thank you so much for the interesting question. Actually, this paper was presented last year on specifics, the results from the institutional and policy structures. But one of the sadly, we have to add sadly at the arm front. Sadly, there is a general pattern of very, very little to no structures, particularly for specifically for women with children. Because one of the from this paper that we just sent for publication or peer review, one of the main reasons why women drop out is family issue. children. So they have to cope with children, and they have to also cope with working in academia or industry, because this also took into account those from industry as well. So when you maybe let's take within academia, no, there's hardly any maybe baby center, crèche or something where you can drop your child, where you can go and stay with your child within the school environment or within the department. So women are forced to just suck it up and get on with the business. And that really does not work well for a lot of women, and that's why dropout is so high and the promotion rate is extremely low. So that is one of the patterns that was observed across across the countries that we took the survey, and sadly that is the outcome. But given that now, we were able to bring up recommendations like at least we need a crèche, a kind of baby environment, or so where the babies can be kept, a play station or something like that, where the babies can be kept there while the mothers go to class or something like when I just fast my own experience when I was in undergraduate, I had 2 children. I can't carry the child into the class while the child is crying. I have to moving out, coming in, moving out like this, and so it's and you can't stay in the hostel with children or with a child. So there are so many restrictions, and since then till now, I have not seen any change. So we need to speak more about this in order to create the change we want.
Yes. Yes. Yeah, I think there's an urgent need for STEM. What you're talking about, Kingsley, in Africa, and I'm really, really honored. I will be honored and happy to be part of any activities you want to take in Africa. So please reach out to us, to me and others in this audience. Okay.
Thank you.
Thank you. I think we are running out of time now. The last talk before the lunch break, at least lunch break in the UK, is by Mohammed. Mohammed is from Somaliland. Are you there, Mohammed? Hello? Can you hear me, Mohammed? Hello? Mohammed, can you hear me? I think you are there. I can see you, but Mohammed? Yeah, I can hear you. Yeah, okay. So are you ready to give a talk? Yeah, I think your talk is going to be entitled Determinants of Birth Interval Duration Among Women in Somalia. Oh, you have some challenge. Okay, there it is. Okay, that's great. We can see your screen. Yeah, we can see your screen. So as I said, Muhammad is also from Somaliland, and I had the opportunity to visit Somaliland this year. There are so many young people in that country who want to do research, but there's not enough mentorship. There's not enough people to mentor them. And I'm working with about, I don't know how many, but I'm working with 24 or more researchers in Somaliland to help them to, reach their dream. And Mohammed is one of them. So over to you, Mohammed. You can begin your presentation. Hello, Mohammed. We cannot hear you. We can only see your screen, but we cannot hear your sound. Mohammed, can you? Hello, can you hear? Can you?
Dear Professor, it's me,
Hello.
Baba. I think he said I have a challenge and he's trying to solve.
Okay. 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 Let him take his time. Okay. We will wait for him. We'll wait for him. Oh, he says he cannot solve due to Microsoft. Um,
Hello.
Hello, sir. Can you hear us?
Yeah, I'm hearing you. Do you hear me?
Yes, of course. We can hear you. Yeah.
Okay.
Okay. So you can.
Okay, thank you, dear Professor Sarales Nagaracha. Another audience, my name is Mohammed. I'm very happy to be here and present my research work. Actually, have you seen it?
Yes, we can see it. Yes.
Okay. So actually, have you seen?
Yes, we can see it. Yes, you can continue your presentation.
Okay. So actually, this study is about duration of breath interval and its predictors among women for childbearing age in Somalia. So this study employs survival analysis. So
Yes.
So here's the outline of presentation. Actually, we will look at the background and also objectives. Second, methodology, and also the results. In our results, we will look here the prevalence of short breath interval and median duration of among women. Also, we will use here a Cox proportional hazard model, which we look at demographic factors, socioeconomic factors, and health-related factors with the duration of breath interval. Also, we will employ multivariate analysis. So in this section, we will use several parametric models such as Webull, Gombart, and also we will look here at the recommendation and conclusion. So when we are talking about breath interval, it's about duration between consecutive life breath, and it's a crucial aspect for reproductive health and also for reduction of maternal mortality and child mortality. So optimal breath spacing generally recommended as at least 33 months between births for WHO. So it's vital for minimizing the risks associated with the closely spaced births. So when we are talking about breath interval, so when we are talking about the breath intervals, there's a short breath interval. Actually, I'm so sorry for the time of Salah, so I'll try to continue. So short breath intervals are linked to Hello, do you hear me?
Yes, we can.
Okay. So short breathing interval are linked to range levels of maternal health and also increased to hemorrhage, anemia, fractures, even for children can contribute to low prematurity and also still birth, malnutrition, and increased infant and child mortality. Globally, this challenge exists. Mostly they are based on a woman experience at the highest in developing countries. So in sub-Saharan Africa, it's are almost 59%. In East African countries, 44%. And women Uganda is about 52. And so on. While globally, short period interval has shown some decline over time, it remains a significant challenge. In Somalia, negative maternal benefit mortality is 150 per 1,000 litres, and maternal rate in Somalia is 692 per 1,000 litres, which is the highest countries in the world. A decrease in maternal, baby, and child mortality is a contingent of family planning. So, according to 2020 statistics from DHS Somalia, temporary contraceptive treatment in Somalia is still low at 1%. Even 9% of women left choose family planning. So here, our objective study is to analyze the duration of breath interval and its predictors in the woman. And these are the specific objectives to determine the median, to determine demographic factors, socioeconomic factors, and also health-related factors. The last objective is to develop or employ survival models for identifying factors associated with the duration of breath interval in Somalia. In method section, the data is DHS also. The study population is reproductive women aged 15 to 49 who have had at least one birth for the last 5 years. The sample size is 7,683 women. So in our study, we will employ univariate analysis, and also we'll use parametric and also parametric models. So we'll continue in our findings. We found that our study, 53% of the women are facing short breath interval, which means they will give another woman less than 33 months. And this is a Kaplan-Meier survival estimate, which showing the median age of a woman is give birth is 31 months, which is far apart the recommended WHO, which is 33 months. So this finding provides important measure of the central tendency in the distribution of breath interval and serves a key reference point reproductive behavior in the population. This is bivariate analysis, which shows associated predictors, so those who are green colors are associated factors. Here, demographic factors, when we are employing Cox proportional hazard model. So this shows factors associated with a significant lower hazard includes maternal age, particularly for women aged 35 years and older. Also, nomadic residence, which was linked to reduced hazard compared to urban settings. And also marrying before the age of 18 was associated significantly lower hazard of short breath interval. Also, there are certain factors found to significantly increase the hazard of short breath interval, such as geographical locations. Women in Huron and Lower Chuba regions experienced higher hazard compared to those in other region. Mostly it's Burma. I know that he visited our RLS this year. In terms of education, women with the primary education had elevated hazard related to higher education. Even women with who had born 5 or more children had nearly double the hazard of short breath interval compared to those fewer than 5 children. Here's a socioeconomic factors. Actually, in this section, as shown the table, all quantiles demonstrated significantly higher hazard of experience short breath interval, except the lowest. So when the wealth increases, they are experiencing short breath interval. In health-related factors with the duration, when we see this, it's related to duration of breath feeding compared to women who have not stopped it. Women who were still breastfeeding experience statistical significance hazard for experiencing short breath interval. Also, even more pronounced has significantly associated was observed with the survival status whose child was died, presumably the previous child is set for a replacement. This indicates that when the mother faces her child was dead, she is expecting to get another pregnancy. So they have a 3 times higher. So here we are combining 5 models, which are logistic, generalized, gamma, Weibull, exponential, Cox proportional. So when we are selecting the best model, we are looking the lowest AIC and BIC. So the best model that fits this data is log logistic accelerated failure model. So here, the output of the best model, which is a log logistic accelerated failure time, which indicates that positive relationship was observed between a mother's age and the infant compared to mother's age 15 to 19. Also, breastfeeding status was a powerful determinant, with currently breastfeeding mothers experiencing time to infant compared to those who had stopped. Also, the residence in the bladder associated with 14% longer to the infant relative to the outer region. Several factors were found to significantly shorten or decelerating the time to infant, such as the death of child was linked to infant, such as the facing short breath interval compared to women, that child was survived. Also, the maternal education also played with the higher education attainment corresponding to short breath interval, and also having 5 or more children associated with 7 short interval. So the recommendation in our study is based on the findings of the study. The following recommendations are proposed to improve maternal and child health outcomes in Somalia through better birth spacing. First, strengthen and promote breastfeeding programs. Second, enhance and target family planning services. Third, integrate health education with the family schooling. Fourth, to develop region-specific health strategies. Actually, here is our conclusion, which concludes the prevalence exceeding 50% and median interval falls or lower than the recommended. The key predictors of short breath interval were identified: lower maternal age, the death of preceding child, short breathing duration, and having some. Hello, do you hear me?
Yes, yes, thank you, thank you. Yes, we can hear you. We can hear you.
Okay.
Thank you for your talk, Muhammad. It's really it's something that is of great value for your country, Somaliland. So it's something you should be proud of. So any questions for Muhammad from the audience? Hello, guys. Hello. Do you have questions for Muhammad on his talk?
Yes, I have a question.
Okay, Louis, yeah.
So, thank you for your talk, first of all, Mohammed. In your recommendations, when you talk about strengthening and promoting breastfeeding programs, enhancing, etc., should this be via the government of Somalia trying to promote this, or should non-governmental organizations or charities try and help females in Somalia strengthen and enhance these things?
Can you repeat me again?
So my question was, for the recommendations, do you think that the government of Somalia should take take the forefront, or should other independent parties try and help women with these problems?
Actually, they are trying to help family planning programs, and they are still not enough to reach or access all the regions in Somalia. Some for a security issue, and also some of lack of funding programs, and some challenges in cultural and religious aspect in family planning issue.
So the main, sorry, I did.
Yeah, go ahead, Louis. Louis, go ahead.
So the main problem would be the funding aspect for the government to try and help these programs, if that's what I understood.
Yeah.
Okay.
Yes.
Okay. Thank you very much.
Okay, thank you.
Mohammed, are there like NGOs, like non-governmental organizations that are helping the population with this regard?
Actually, the non-governmental organizations, they are trying to do this, but mostly they are limited to funding projects. They are a timely project.
Okay.
So if they can start 1 year, but after that 1 year, there's no another sustainability. So still, even in our societies in Somalia, when we are talking about the family planning issue, they are hesitating to use, or even they don't have some background knowledge. Even when the communities or non-governmental organizations are coming to rural areas or in urban areas, they do not explain morely in a indigenous knowledge, like through messages from religious leaders or leaders in a community. So when they are coming to some place, they are coming alone.
Yeah.
So when they are conducting some awareness or some community programs, it's better to be part of the communities. Actually, those who are elite, those who are leaders, those who are voters, that communities, they are more confident.
Sure. Exactly. Yeah.
So that's some kind of challenges.
Yeah, we understand. Yeah, I mean, I think all this care needs to be taken by African themselves, not depend on. I mean, we cannot depend on the outside world to help us. I mean, that's part of one of the reasons for this workshop is to help for Africans to help their fellow citizens. I mean, that's the purpose of this workshop. I think that goes with everything. That goes with not just research, but also with what you're talking about in mental health and childcare, etc. So we cannot depend on the outside world. I mean, we need to help ourselves. So that's really important. And that's a message that we need to take to everybody, everyone in the continent. Yeah. Okay. Any more? Any more questions for Mohammed, please? Okay. So it's now 30 past 12 in the UK. So it's lunch breaks. We're gonna have a lunch break. So we will start again in 1 hour time. That is 30 past 1 UK time. So can you? So you can. So wherever you are, I don't know exactly the time at your places, but take a lunch. Take a break for 1 hour, and we'll be back again. Okay. Thank you. This one. Test. Hello, everybody. while we are waiting for people to join. Let's give a few more minutes. Well, Emmanuel, it will start in a few minutes' time. We are waiting for the speaker. We are waiting for the speaker to come. I just sent him an email. I don't know, maybe he's busy with something. Oh, there he is. Okay, Victor is here. Hello, Victor. Can you hear us? Victor?
Oh, sorry, I just joined.
It's okay. Let me introduce you to the audience. So Victor is from University of Zambia in Lusaka. He's a good friend of mine and a collaborator. We knew each other we've known each other for 3 or 4 years now. Victor visited Manchester about 2 years ago. He was here for 3 months. I visited Victor in Lusaka in this year, about for 1 or 2 weeks. He is a very extremely productive. I mean, he and I, in the last 3 or 4 years, we've known each other. We published like more than 10 papers together. Victor did his PhD at the University of Waterloo some time back. But he started becoming productive as I got to know him about 3 or 4 years ago. He has really been inspired, like I am, okay, to contribute to the continent. Okay, Victor, I will let you present your talk. Yeah, okay.
Okay. Just share the screen. Are you able to see my screen?
Yes. Yeah.
Thank you very much. Good afternoon, everybody, and good morning to some of you, depending on where you are. So the title of my presentation this afternoon is Logarithmic Method of Moments Estimators for the Frechet Distribution. And this is the outline of the presentation. I'll begin with a brief introduction. Then I'll talk about common estimation methods for this particular distribution. Then I'll go on to talk about the proposed closed form estimators, then look at the large sample properties, make some comparisons with some estimators, and then a conclusion and some references are going to be given. So just a brief introduction about the Frechet distribution. This is a special case of the generalized extreme value distribution. And it is basically actually the inverse of the Weibull distribution. It is commonly used to model heavy-tailed data, and it has wide applications in many fields of study. Some examples here include extreme weather events, accelerated life testing, change point detection, athletics, random forest regression, and many other areas. Here we have only a few references, otherwise it has a lot of applications. So this is the Frechet distribution. It is a 2-parameter distribution. So we are going to denote it here by FR with parameters alpha and lambda, and that is the PDF. It is defined only for positive values of X, and the parameters alpha and lambda are also positive. And it has a nice CDF, cumulative distribution function there, nicely closed. And the RF moment as well, except it depends on the gamma function and is only defined when R is less than alpha, where that is a gamma function defined by that integral there. In terms of parameter estimation, a number of researchers have proposed and compared different methods of estimating the parameters. Some of them here include Bayesian estimators, as proposed there by NASA in 2015. And then a number of comparisons have been made. Here we have Malek and others in 2017, comparing maximum likelihood, uniform minimum variance, least squares, weighted least squares, and percentiles estimators. And then we have Mohammed in 2017 as well, comparing least squares, L moments, Bayesian, and maximum likelihood estimators. And then we more recently, in 2020, we have another comparison there by Coelho and others, comparing maximum likelihood, maximum product spacing, L moments, and moments estimators. So here we are going to review some of these commonly used estimation methods. Mainly, we restrict ourselves to these three, which include maximum likelihood estimation, method of moments, and L moments. So if we have a random sample of size n, that is drawn from the Frechet distribution with parameters alpha and lambda, the maximum likelihood estimators are solutions to these two equations here. And as you can see, we are not able to obtain closed form solutions. So we need the iterative procedures for us to be able to solve these equations. But given that we are able to find those two maximum likelihood estimators, alpha hat and lambda hat, for alpha and beta, Ramos in 2020 showed that the maximum likelihood estimators converge in distribution to the true parameter values, and variance covariance matrix, which is the inverse of the Fisher information, as n goes to infinity, and the Fisher information is given by this matrix here, which, as we can see, depends on pi, as well as on the Euler constant, as well as the log of lambda. So this Euler constant here, of course, can be written in terms of the digamma function. And we can easily show when we invert that Fisher information matrix that we get this expression here. And then now we are going to look at the method of moments. So the method of moments basically compares population to sample moments. So the first two population moments are given by these two expressions. So basically, we simply put the R as 1 in the previous expression for the rth moment to obtain the expectation, the first moment, as well as putting R is 2 to get the second population moment. And then we can equate these population moments to the sample moments. So that will give us the solution. The solution to these two equations, we yield the method of moments estimators. Again, as we can see, we do not have closed form solutions to these two equations. And apart from that, we also notice that we only get solutions under the restriction that alpha is greater than 2. So we have a restriction on alpha. And then when we move on to the method of L moments, so this method here was introduced by Hoskin in 1990, and it is basically solving the equation of estimating parameters by equating L moments sample moments to population moments. So the first two sample moments are given here as L1, is basically the mean, and then L2, which is a function of the mean and the order statistic. And in 2020, Ramos showed that the first two population L moments are given by these two expressions here. So they depend on the gamma function, and equating these population moments to the sample moments earlier showed there leads to these two L moments estimators for alpha as well as for lambda. So we get closed form solutions, except again, we have another restriction in that for us to be able to get these, we need alpha to be greater than 1. So what is the proposed closed form estimator here? As the title says, basically it is based on making a transformation on the original random variable x, getting a log of that, and then obtaining moments estimators for that. So if we have our original random variable x, which is a Frechet distribution, and then we make a transformation by getting the log of that, and then the new PDF is given by this expression here with the change that instead of it only being positive, now it is defined over the entire real line. And then we can easily get those two population moments given by these two expressions in 3. So they depend on log of lambda as well as on this Euler constant here, as well as pi. And then we can use the method of moments, equate these two population moments to the respective sample moments. And so this is what we have here. And then we solve for alpha and lambda. And when we do that, we get these proposed closed form solutions here. So basically that is how we arrive at those proposed closed form estimators. And we can show that those estimators are strongly consistent for alpha and lambda. And we can also show that they are asymptotically normally distributed. They converge to the true parameter values as n goes to infinity with a variance given by this expression here. So when we look at this expression here, it depends on log of lambda, it depends on the Euler constant, it also depends on a Paris constant. And what we do here is make a comparison between the different methods of estimation. So as we have seen already, the method of moments estimators only exist if alpha is greater than 2. The L moments estimators only exist if alpha is greater than 1. The maximum likelihood estimators exist for all values, possible values of alpha and lambda. The only challenge we have is that they are not closed, are not in closed form. And so for the purpose of comparisons here, we are only comparing maximum likelihood estimators to proposed estimators as the only two that do not have any restrictions on alpha. So if we were to look at the asymptotic variances, that is comparing the asymptotic variances of the maximum likelihood to the proposed estimator, we get this ratio here for all values of alpha. And this ratio here is always less than 1. And similarly, if we are to get the asymptotic variance of the maximum likelihood estimator to the proposed estimator for values of lambda, we also get something that is less than 1, meaning that the maximum likelihood estimator is more efficient than the proposed estimator, that is as n goes to infinity. But the question probably might be, how about for small values of n? And that's a comparison that is of interest to us here. So that comparison is done through looking at the bias as well as the mean square errors through simulations. So for example, here we pick some small values of alpha and lambda, and then we do simulations for alpha and lambda, and then we compare the biases. So what we notice here, so we are changing the sample size from 10 to 25, and what we notice here is that our closed form estimator here has a smaller bias compared to the maximum likelihood estimator. And similarly, when we look at the mean square errors, we get more or less the same picture. The mean square errors for the maximum likelihood estimators are quite large compared to the mean square errors for the closed form estimator. And here we have another set of values of alpha and lambda. And generally what we see here is that the biases here for the mean square errors are even a little bit bigger compared to what we had in the previous set of values. And we have the same picture even when we look at the mean square errors. And we took another set of values of alpha and lambda. And here we actually see that the difference is even getting larger in terms of bias, as well as in terms of mean square errors. So in short, what we have done here is we have proposed a closed form estimator that is based on a log transformation of the original random variable for the Frechet distribution, and then applying the method of moments. And what we take from here is that our proposed closed form estimator, unlike the moments estimator or the maximum likelihood estimator, takes closed form estimators, the form, and the importance of closed form estimators cannot be overemphasized. And we will also notice that the proposed estimator is valid for all parameter values, and like as we have seen for the method of moments estimators or the L moments estimators. And then apart from that, for certain combinations of values of alpha and lambda, we have seen here from the graphs that our proposed estimator can leads to better efficiency. So these are the references. And if you are interested in reading more about this, that work is contained in this paper here. Thank you.
Thank you, Victor. Thank you. Any questions for Victor? Hello. Questions? Anyone has questions for Victor? Yes, go ahead. Emmanuel, you can ask the question. Emmanuel?
Yes, hello.
Yes, go ahead.
Hello.
Yes.
Are you hearing me?
Yes, of course, we are hearing you. Yeah.
Yes. I first of all, I want to thank the presenter for the wonderful presentation. I am so happy to listen to the presentation. I'm called Emmanuel Aling from Uganda.
Mm-hmm.
Yes, I am happy that from your talk you showed that maximum likelihood estimation is more efficient for large samples. I wanted just to inquire if you can do also some estimations using the Bayesian methods and how you can apply them to you to come out with estimation. Thank you. Though I'm still new in the field, I will be grateful. Thank you.
Okay. So actually, I think I gave you some references somewhere at the beginning where the Bayesian approach was used. Let me just get to that. So I think if you look for this paper here by Nasra in 2015, there are some Bayesian methods that are applied to this same particular problem. And if you also look at this paper here by Mohammed in 2017, there is actually a comparison that is being made there, comparing Bayesian estimators to the other estimators.
Okay, thank you.
All right. Thank you. Thank you, Emmanuel. Any other questions for Victor? Hello. Anyone with more questions for Victor? Yes, of course. Of course, we will share the presentation, and all the talks are currently being recorded. So we will share the videos of all the talks later on, later today and tomorrow. Thank you. So Victor is an example of a person in, trying to do research in spite of the difficult condition, in particular the teaching load as in many other countries in Africa, the teaching load in Zambia is quite high. And the number of students is also pretty high. So in spite of all that heavy teaching, and Victor is determined to do good research, and everybody in Africa should be proud of that and try to emulate what Victor is doing. Okay, the next talk. Thank you, Victor, for your wonderful talk. The next talk.
Thank you.
The next talk is from Abdi Rahman from Somalia. Abdi, are you here? Abdi, hello. Abdi Rahman Omar Ali, are you here? Not sure if he's here. Abdi Rahman Omar. Mm-hmm. Hello. Hello. Is there somebody called Omar Ali at this? Hello.
I don't think there's someone because I can't see such name on the participant list.
Mm.
I don't think a person is available now. We can move to the next speaker, and then maybe by that time he'll have.
Okay, Louis, are you? Can you speak now, Louis? Louis,
Yes, yes. Just give me 1 second and I'll be ready.
sorry for the change of
No
time,
problem.
but the person at 2 o'clock does not appear to be here.
Yeah, that's not a problem whatsoever. Um,
So Louis is an undergraduate student here in Manchester. He is originally from France, but he is somebody very keen to do research. So he has been working with me on writing papers to do with African data. It's been an inspiration to work with Louis.
um, okay, share. Can everyone see my screen?
Yeah, can you make it larger if possible? If it's possible, yeah. Okay, that's
Yes.
fine. It's better now.
Yep.
Yeah, that's best. Yeah, that's good. That's great.
Okay. Just, yes. So I'm going to present a paper that I co-wrote with Sarah Lees over this summer about the seasonal effects in the Nigerian stock market, where we use statistical testing to try and analyze the price gaps in the Nigerian stock exchange. So the motivation behind the paper is that Nigeria is an emerging economy, not just in Africa but also in the world. So we can expect potential increase in investment in the country and in the market as a whole. So any research that we do now could be built upon later on when Nigeria could rivalize with more distinguished and developed economies. Currently, there's quite a lack of research in price gaps in the Nigerian Stock Exchange. I'll come to defining what a price gap is in the next slide. So by having a look at how price gaps act in the Nigerian Stock Exchange, it can give us an idea of what we can expect and how we can try and not manipulate the market, but try and understand the market better. Why the market behaves the way it does, and what we can do to help the market and Nigeria's economy as a whole. Um, it's also the data is readily available. It's quite easy to find on the internet, so it's really important that we do the research because we have the data for it, and the consequences can be immense. So therefore, it's a great reason to do said research. We're concentrating here on the NSE 30, not the NSE All Shares Index. NSE 30 is just the top 30 publicly listed companies. So it's the best performing companies in the stock market and most liquid companies in the stock market, which is more important to analyze than the All Shares Index, which just tracks all of the stocks and shares in the Nigerian Stock Exchange, because not all the companies have shares to sell at any available moment. So therefore, any effects that we might be able to see, we might not be able to act upon. Why we chose to look at seasonality in particular is because there's repetition in seasonality. We can use the past to infer what we can expect from the future. And as I said before, the data is readily available for historical past data. So defining price gaps. So the stock market in every country opens and closes, but sometimes the opening price and closing price differ, and that creates a price gap. So numerically, a price gap is gap percentage of a price gap is the opening price minus the previous closing price divided by the previous closing price, and then multiplied by 100 to get it into percentages. Why are price gaps important? Well, the efficient market hypothesis is a major hypothesis in investing, economics, and finance. It assumes that the market reacts instantaneously to all data that is readily available. It assumes that there's no secrets. All parties know data. All data is readily available, and all parties know the data and act within reason, which isn't the case in real world scenarios. So the fact that the market closes and opens, that there shows that the market can't instantaneously react to any information that comes out between the closing and opening times, which means any differences in those prices is an anomaly and hence is ground for exploitation of the efficient market hypothesis. So if we do an initial plot of the gap percentage over time, then we see that there's no clear pattern. If we look at the years, every year, maybe from 2012 to 2014, there's more activity in price gaps. However, it's quite spread out. Particularly from 2020 to 2025, it seems quite consistent. There is one anomaly here, which we do take into account. The QQ plot shows that a lot of the data is non-normal. It shows that the data is compressed around zero because this takes into account non-null price caps and the null price caps as well. And the data that we're working with, only a third of the price gaps are different from zero. As we can see in the histogram of all the price gaps, a lot of them are concentrated at zero. It's important to note that this is a zoomed in version, so the number of zero price gaps exceeds 500. Additionally, when we have a look at the histogram of non-null gaps, we can see that there's a slight skew to the right. We can clearly see it's not normally distributed. Therefore, we use non-parametric testing in the following tests. An important thing to note: the data that we use is from 2012 to 2025, so it uses a lot of past historical data, which we use to infer future data. If we start with a weekday seasonality, we plot the gap occurrences. So this is non-null gap occurrences by a weekday. We see that the non-zero gap occurrences seem to be quite evenly spread out, and by using three different non-parametric tests, we get a p-value that exceeds 0.5, which is much greater than 0.05, sorry, meaning that we can it's safe to say that hypothesis 1 is true. We then go on to hypothesis 2, where we test to see if the average of each weekday price is different across weekdays. Because if they are different, then we can see if one weekday is more positive or negative than the others. And here, by looking at the bar plot of the averages, we see that Monday is the only positive average, and Tuesday and Friday seem to have quite a strong negative average. However, when we use parametric tests to see if Tuesday and sorry, I've skipped ahead just a bit too much. The effect size. If we use the effect size measured by the epsilon squared from the Kruskal-Wallis test is only 0.006, sorry, which means that although although the difference is significant between the weekdays, it doesn't necessarily mean that there's grounds for exploitation in the real world. However, it might still be interesting to see why there are differences between the weekdays, even though we might not be able to exploit it in terms as an investor. So if we have a look at the averages of the price gap, we see that Tuesday and Friday are quite a bit more negative than the other days. However, there's Tuesday is the only significantly negative one, and this is most likely due to the fact that Friday has the outlier that's seen before. This outlier here is on a Saturday, and it has a value of minus 5.78, which completely skews the average for Friday, and hence why the t-test and Wilcox test doesn't return as a significant for Friday. So with this being said, we can now try and compare the distributions of the weekdays to try and see if that might give us a better telltale sign of why exactly some weekdays are more or less negative, have more or less price gaps. So if we have a look at the plots, the initial plot of a distribution of that percent by weekday doesn't tell us much, but if we zoom in, we see that Tuesday seems to have this strong line that tends to be more negative than the other weekdays, even though it doesn't seem to be that pronounced. Uh, this hence leads to our hypothesis 4. Price gaps distributions differ from weekday A to weekday B, and by weekday A and weekday B, I mean that it represents Monday and Friday, for example. So distributions of Monday and Friday differ, or the distributions between Tuesday and Thursday differ. Um, when we by using extensive pairwise comparisons, we find that there's a difference between Tuesdays and Fridays distributions, and also between Wednesdays and Fridays distributions. It's all well and good to know this, but we don't know yet why there's a difference. And if we have a look at the averages of Tuesday and Friday, they're not that much different, and as well the averages of Wednesday and Friday don't seem to be that much significant because Monday's positive and Friday's negative, but there's not a difference in distributions there. So here we have a look at the number of positive, negative, and null gaps between each weekday. And if we use the chi-square test, G-test, and Monte Carlo multinomial test to see if there's a difference, we see that the downward gaps present a significant change among the weekdays. And if we have a look at them individually, we do notice that Tuesday and Friday, Tuesday, which has the most downward gaps of 134, is significantly different to Friday's 79. And again, Wednesday's second highest with 107 is also significantly different to Friday's 79. And if you use the chi-square test, G-test, and Monte Carlo multinomial test with just each of the two weekdays, we notice that there's a difference, significant statistical difference between the amounts, which could hence lead to the difference in distributions. We can also have a look at the variances of each weekdays. As we look at the plot, Monday through Thursday seem to have a quite similar amount of variance, whereas Friday has a high amount of variance. Through statistical testing, we don't see any significant differences between the weekdays and their variances. Friday's large amount can be due to, as said before, the extreme outlier. So sorry. Some of the links we can try and do towards other papers in Ayunko and Johnny 2020, their paper titled "A New Look at Weekday Effects of Stock Prices in the Week Form." They concluded that the NSE, Nigerian Stock Exchange, is efficient in the week form. using quite recent data, 2018 to 2019, sorry, which does help strengthen our argument of the exploitation of the potential exploitation of the efficient market hypothesis. Um, because when it says that the NSE is efficient in the weak form, it means that it isn't completely efficient. So there's gaps in the efficient market hypothesis in the NSE that could be grounds for exploitation, such as the price gaps. Also, in RGDD in 2008, their paper notes that there's a significant positive average return on Friday in Nigeria during the trading day, which could potentially explain the lack of downward gaps and the excess of upward gaps on Friday's anticipation. Um, just to make just to show that that makes some sort of sense. Um, if we're expecting the stock prices of the NSE to go up during the trading day on Friday, then from Thursday to Friday, there won't be it wouldn't make sense for the price of the stock prices to go down from Thursday's close to Monday to Friday's opening, if we know that Friday January tends to go up as a whole, um, which also explains the amount of upward gaps in anticipation of the increase of the stock prices during the trading day of Friday. The turnaround effect, a correction of Monday's sentiment, really could explain Tuesday's significant negative average. The Monday effect is effect where, because the stock market is closed over the weekend, there's much more volatility between Friday and Monday's price gap, which means that Tuesday's price gap, the close between Monday and Tuesday, could mean that there's a lot of adjustments between Monday's trading day, and hence could mean that Monday was much more anticipated to be positive, whereas it didn't reflect the current price on the stock market, and hence during the close of the stock market, it corrects itself and becomes more negative on the opening of Tuesday. If we have a look at the monthly seasonality, we see that the distribution of non-negative gap occurrences by month is much more unevenly spread than for weekdays. If we come back and have a look at the weekdays, the 5 weekdays are much more even than the months here. So this leads us to hypothesis 5. Price gaps are unevenly spread throughout months. And using the same statistical tests as we did for hypothesis 1, we find that in this case there is strong evidence to show that the non-null gap occurrences are uneven throughout the months, which could be grounds for exploitation as well, which leads us to hypothesis 6, where we test to see if the amount of non-zero price gaps in month A, such as January, February, March, April, May, are significantly more or less than expected. So the table that we see underneath hypothesis 6 is the only statistically significant results that we get, where we get that March, April, October, and December vary significantly from the mean of the gap occurrences per month. In this case, it shows that March and April have statistically more gap occurrences per month, whereas October and December have statistically less gap occurrences than the average month. This means that there's more, it could mean that there's more volatility for March and April and less volatility for October and December for the stock prices, because maybe there's less information, maybe there's more information. We'll see later on why we can try and explain that. If we now have a look at the averages of the price gaps per month, we notice that it again seems very uneven, such as the number of gap occurrences per month. Using statistical tests, which are non-parametric, we find that p-values are significant. However, UN's test with 1% trimming is significant, whilst UN's test with 10% trimming isn't significant. This could mean that there's a low amount of price gaps that might have skewed the data to show significance for the whole data, whereas 90% of the data isn't isn't statistically negative. Um, this might be important. However, still with 10% trimming, the p-value of UN's test was still 0.17. So we can still assume that the averages of price gaps are different across months. Something to note here is when we measured the effect size of a Gruskin-Wallace test, we get a measurement of 0.03, which is much greater than that of the weekdays, but still might not be significant enough for practical use for investors. But it's still important to note, nevertheless. Yeah, so if we continue with the chronological order that we did for weekdays, we now have a look whether the month A's price gap is the average of month A's price gap is significantly positive or negative. Here, as you can see in the table, the February very is quite significantly statistically different, which means that February here significantly positive. We also have that April is significantly negative, and that November might be significantly negative, and December may be slightly significantly positive. If we continue as we had a look with as we did for the weekdays, and we compare the distributions of the months, here we can see more of a difference in the box plots. February seems to tend to be more positive. December seems to be more condensed as July, August as well. June seems to be more spread out, such as March, which leads us to hypothesis 9. Price gaps distributions differ from month A to month B. And using a pairwise comparison, we see that the distributions of February, April, February, May, February, June, February, and November differ, as well as distributions between April and December differ. So if we have a look at the upward and downwards null and total gaps of the months, we can see that February has the lowest, with December at second lowest. October has quite low non-null gaps. It has very high null gaps. April has the highest positive gaps, highest null gaps, downwards gaps, sorry. And as we can see with the chi-square test, G-test, and Monte Carlo multinomial test, the differences between the downward gaps are very significant compared to the upward gaps and null gaps, which leads us to believe that the differences in distributions of February, April, February, May, February, June, February, November, February, May, June, and November could be explained by the fact that February has 18 downward gaps, whereas the other ones have quite a normal amount. This is also proven through in the paper, we actually do tests to comparing each month to the other month's downward gaps, and we see that this is one of the main causes for that. We also have a look at the variances in the paper. However, that doesn't it doesn't show much to conclude that the variances are one of the main reasons why the distributions differ. If we look at the links for the monthly seasonality, as we did for weekdays, we have a look at one of the papers written by IID in 1998. It demonstrates that there's not much evidence of there being a January effect in the Nigerian stock market, despite finding one for the Ghanaian stock market, which shows that there's differences in African stock markets themselves. The January effect is the historical pattern in the stock markets where prices show a notable uptick during January. We haven't found this in our research, so this paper to support our findings and the fact that there's a lack of January effect. The statistically negative gaps in April and May could reflect fiscal year adjustments or tax-related selling pressure, as suggested by Intim and Osei in their paper, The Impact of Corporate Board Meetings on Corporate Performance in South Africa. So, although their paper was based on South Africa, it does show it's not limited just to South Africa, as we see in our case for Nigeria. It's also strengthens the argument. It's also important to note that April, the large effects that we have in April could be linked to the fact that it's close to the end of the second quarter of corporate companies. So it could be in anticipations of the results of companies' second quarter, end of second quarter, and etc. If we have a look at the links to other countries, Plaston and I have to look at the US market, which found strong weekday patterns, which we didn't find much of a weekday pattern except for Tuesday, which was significantly negative, which could be an effect of a weekday, of a Monday effect, but we didn't find a Monday effect ourselves in our research of Nigeria. Lebeau, in 2018, found that there's a pre-holiday effect in Egypt, Tunisia, and Nigeria, and they investigated the month of the year effect, which both support the findings of calendar effects in the Nigerian Stock Exchange. Van Berg and Van Zyl, 2025, found that for global indices, there's no strong evidence for the reversal of gaps towards the previous close, meaning that they tend to fill. This means that if for the indices that they had to look at, the Nasdaq and the DAX, during the trading day, if there was a positive or upward price gap, then the price of that index would continue to grow. So the movement of the price gap kept on going within the trading day. And I'm coming close to the end. The Eurostat and ONS, which is a European committee, provide seasonal adjustment guidelines highlighting an infra-annual effect in financial time series, including weather and institutional cycles for European countries. In the paper, we also have a look at the differences between seasons. In today's presentation, I didn't think I would have time to talk about the seasons, so I didn't mention it. But spring has many more amount of price gaps compared to the other seasons. And this is most likely due to the weather phenomenon in Nigeria, which could create instantaneous news information, which could harm companies, such as companies that are dependent on farming during a tornado or a dry season, etc., etc. And this concludes the end of my presentation. Thank you very much.
Thank you, Louis. That's really comprehensive study of Nigerian stock market price gaps. Yeah, I mean, yeah, it's interesting. Like you find all these differences between weekdays and differences between months. It'd be good to know why. I mean, I know there are some Nigeria Kingsley is from Nigeria. I guess Kingsley, would you know why? Like, for example, the price gaps differ between, for example, is it Tuesday and Friday, for example, they differ, and so on. Would you know why?
Actually, I was cut. I'm surprised by the results, and that question just kept on ringing in my head. Like, yes, I'm doing a work on NSC as well, but that's on an actual level. And I was thinking it would be nice to know if have physical interpretation of why Tuesdays and Fridays seem to have those negative returns. That would be something that would be so interesting to study about, which I may be interested in doing. So I really appreciate and I love the work that has been done by Lois. It's really illuminates the different aspects and the disaggregation into weeks and monthly variances is quite interesting. So I don't have any interpretation, but a physical interpretation will, and if it is being compared with other emerging markets across Africa, um, then that may now either isolate or show the similarity in terms of maybe the physical occurrences, or maybe at regional level, West Africa, East Africa, and all that. So that may also highlight specifics about the characteristics of that Tuesday and Friday dynamics. But I'm sorry, I don't have any very solid reason to give.
Yeah,
I could
yeah.
potentially try and explain the difference between Tuesday and Friday.
Mm-hmm.
So maybe I didn't have time to talk about it in the talk, but Tuesday, um, the amount of negative gaps on Tuesday could be due to the Monday effect, which we don't see, but it could be a correlation with the Monday effect, where Monday might have, after the weekend, the trading day in Monday went so high that it will overshows what it should have done. So when the market closes, it goes back down to what it um probably what it should cost. And on Friday, one of the papers shows that the during the trading day, the index prices go up during the trading day of the NSE 30. Therefore, if we know that the price tends to go up on Fridays, then during the close of Thursday and Friday, it would be irrational to think that it would be irrational to suppose and irrational for the prices to go down before they go up. Because if we know they're going to go up, then it's logical to think that they go up. It's logical to buy, for example, it would be logical to buy now and then sell when they're already up during Friday as a supposition, if that makes more sense, but it might not make complete sense.
It makes some sense. It's just that it's a theoretical explanation or an academic explanation. Well, I think it would be also good to add a physical explanation. For example, what takes place physically on the ground in the exchange, in the Nigerian Stock Exchange on the Fridays. Maybe something like a sentiment analysis around those days may give some, may illuminate more on that. And it does that, is that specific or unique only to the Nigerian market, or does it also reoccur in other markets across Africa or within that region? Like since Nigeria is in West Africa, does it occur? So that physical interpretation will be quite beautiful. So that it can now be linked to things like policy. You can now have some concrete policy recommendations based on the physical activities of the market. So that's where I think I'm coming from. Yeah.
Yeah, I think Kinsley is making a great point. I think it would be good to have some kind of a physical interpretation, and also like how things differ between different regions of Africa. I mean, Louis doing the same thing for South Africa and Morocco. So they are in three different directions. Like Morocco is north, Nigeria is west, South Africa is south. So we can see if there are differences in the behavior for the three different regions of Africa, or are they the same? So I think, uh, I think it'd be good to look at it. Yeah.
Yeah, thank you very much, Quinsley, for your comments. They're very, I took notes down and they're very useful. Thank you.
Yeah. Yeah. Any other questions for Louis from the audience?
Louis, will it be possible to seek for that data, the data you're using? Will it be possible for, like, maybe if I want to use it for other analysis, can I get data from you, that the specific data you used for this analysis?
Yes, yeah, I can send it to you if you want. Yeah, no problem.
All right, thank you so much. Thank you.
No problem.
Yeah, thank you. Or you can just post it on the chat or something. If you know the link, Louis, you can just post it on the chat room, or I can give you the email for Kinsley later on. You can send it to her.
Like, I can send you the link on the website where I found the data. I can just send you the data itself in an Excel format
Yeah, I would love the
on
one.
your email if you want.
I would love the one in Excel.
Excel?
Yeah, Excel. Yeah.
Perfect. Thank you.
All right. So thanks again, Louis, for a very inspirational talk, and it's very comprehensive. What you've done is really great work. It's just that we need more interpretation, theoretical or physical or whatever. I think it would be great if we can get some of it. Yeah. Yeah. All right. Let's see if this person is here. Abdi Rahman. Is there anyone called Abdi Rahman here? Abdi Rahman. Seems nobody is here. All right. So if he is not here, I think we can close it for today. And thank you for everyone who presented today. Once again, Hodo, Anotida, Kinsley, Mohammed, Victor, and finally Louis. They all presented great talks today. And all the talks are being recorded. They were recorded. I will present them later on. Tomorrow we have 4 more talks tomorrow. Kwame, Adika, Patrick, and Dima, they will present their talks tomorrow. So the sessions tomorrow will start at the same time as today at 10 a.m. UK time. So please do come, tomorrow. And after the talks, we will have a brief discussion about future plans, right? And if you can also contribute to that, that will be much appreciated. Okay, thanks once again for everybody who presented, and thanks once again for everybody who contributed to the discussions. And thank you for taking your time to present your work. And let's keep working together, and let's make things great for Africa. Okay, so wherever you are, have a good afternoon or good evening or good morning. I'll see you tomorrow. Thank you.
You're welcome. Yeah, take care.
Take care. Bye.
Yeah.
Bye bye.
Thank you.
Thank you.
Thank-
Bye.
Bye.
Thank you, Professor.
Bye bye.
Bye-bye.
Bye. Thank you.
Bye.